Honorable Chief Guest, Guest of Honor, respected patrons, esteemed faculty members, and dear fellow mates. I, Zainab Zahid, research scholar at Interdisciplinary Nanotechnology Center, am honored to stand here and welcome you to this GIAN program titled as a multidisciplinary approach towards drug discovery and delivery being held from 25th November to 29th here at Aligarh Muslim University. This program is itself special because this happens to be the first GIAN program being organized by the Interdisciplinary Nanotechnology Center, Aligarh Muslim University. I again welcome you all. It is noteworthy that AMU was inspired by the academic models of Oxford and Cambridge universities. This noble institution was created with the mission of imparting Western-style education while preserving cultural and ethical values. And it has since bloomed into a beacon of learning and research for over a century. Over the decades, it has flourished into a center of global learning and research. We have been joined as guest of honor and foreign faculty by Dr. Imadul Islam, an eminent scientist with over 25 years of global experience in drug discovery. He serves as a research scientist, Deputy Director Drug Discovery and Head of Pharmaceutical Analysis at King Abdullah International Medical Research Center. May I now please call him on stage. It is noteworthy to mention that the Government of India began GIAN initiative with the aim of allowing direct interaction between Indian students and faculty to gain exposure to world-class knowledge, advanced methodologies, and cutting-edge research trends that might otherwise be inaccessible. Very much in favor of India-abroad collaboration. We have been joined as chief guest by Mr. Syed Ahmed, CEO of Tech Invention. He is a visionary leader in global vaccine development, biologics, and healthcare innovation. He works across more than 30 countries and stands as a testament to his mission of accessible healthcare to millions worldwide. May I now please call him on stage. Accompanying with us is our special guest and Indian faculty, Dr. Mohammad Zainuddin, Senior Director at Jubilant Therapeutics, whose expertise in drug metabolism and pharmacokinetics greatly enriches the scientific foundation of this program. May I now please call him on stage. Morning, sir. We are privileged to have with us an eminent assembly of patrons and faculty whose remarkable contributions have shaped this event to a wider field. We have with us Professor Muhammad Muzammil, Principal, Zakir Hussain College of Engineering. May I now please call him on stage. Next up, we have Professor Vibha Sharma, our local GIAN coordinator and internationally celebrated linguist and scholar. Next, we have Dr. Muhammad Azhar Aziz, Associate Professor at Interdisciplinary Nanotechnology Center. He'll be serving as our course coordinator. May I now please call him on stage. I also extend my heartfelt appreciation to the organizers, Dr. Suala Naseem and Dr. Syed Afzal Ahmed, Interdisciplinary Nanotechnology Center, for their dedication and efforts in bringing this program to life. AMU holds the tradition of beginning its ceremonies with spiritual reverence. To commence this program with blessings, I now invite Akif Dawood, a BTech third-year student, to recite verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الله نور السماوات والأرض. مثل نوره كمشكاة فيها مصباح. المصباح في زجاجة. الزجاجة كأنها كوكب دري يوقد من شجرة مباركة زيتونة لا شرقية ولا غربية يكاد زيتها يضيء ولو لم تمسسه نار. نور على نور. يهدي الله لنوره من يشاء ويضرب الله الأمثال للناس والله بكل شيء عليم. صدق الله العظيم. Translation of the verse. Allah is the light of heavens and the earth. His light is like a niche in which there is a lamp. The lamp is like a crystal. The crystal is like a shining star, lit from the oil of blessed olive tree, located neither to the east nor to the west, where whose oil would almost glow, even without being touched by fire. Light upon light, Allah guides whoever He wills His light, and Allah sets forth parables for humanity, for Allah has perfect knowledge of all the things.
Jazakallah khair Akib for the soulful recitation. As we move forward in today's program, it is now time to honor our distinguished guests who have graced us with their presence. First, we begin by honoring our guest of honor. May I now request Professor Muhammad Muzambil to present the memento to our guest of honor, Dr. Imadul Islam. Thank you so much, sir. Moving ahead, as a gesture of warmth and sustainability, we would like to present potted plants to our esteemed guest. May I now request Professor Vibha Sharma ma'am to kindly present potted plant to our chief guest, Mr. Syed Ahmed. Thank you so much. I will now request Dr. Azhar sir to present the potted plant to Dr. Mohammed Zainuddin. Sir, I again request you to present the potted plant to Professor Mohammed Muzambil, to our principal. Now I request Dr. Kasid Anwar to present the potted plant to Professor Vibha Sharma, our local GIAN coordinator. Ladies and gentlemen, it's time to invite the coordinator of this GIAN program, Dr. Muhammad Azhar Aziz, Associate Professor at Interdisciplinary Nanotechnology Center. He is an expert in cancer research with pioneer work on precision and personalized medicine in colorectal cancer with an integrative approach. He established Cancer Nanomedicine Consortium at Aligarh Muslim University to bring together different departments for conducting cancer research and find translatable solutions for the patients. He has a vast global research experience through working at world-renowned institutions like Case Western Reserve University, Cleveland, Ohio. He was a part of Integrative Cancer Biology Program, National Cancer Institute, USA. His latest award was from the Ministry of National Guard Health Affairs, Saudi Arabia for Best Researcher. I will now request Dr. Mohammed Azhar Aziz to kindly deliver the welcome address.
Thank you, Zainab, for the kind introduction. First of all, I would like to welcome all the dignitaries on the dais and off the dais, especially Professor Muzammil Saab. He is the current principal, and we also have the former principal with us. And they are the visionaries who are knowing that what is the importance of these multidisciplinary efforts. And we also have Professor Vibha Sharma. She is a well-accomplished linguist, and she has recently got her award for the best teacher. And definitely, we have got our chief guest, Syed Ahmed Saab, who is here after a long journey, which was marred by flight cancellations and flight disruptions as well. So I sincerely thank you, sir, for being here. And Dr. Imad Islam coming all the way from Saudi Arabia. And, you know, traveling these days is becoming difficult with a lot of flight disruptions. So thank you for being here. And Dr. Zainuddin, he is our industry expert and will be with us for the rest of the course. So I wholeheartedly welcome all the participants here who are going to be the best benefactors, learning from the faculties and learning about the topic, which is very relevant for today. I think it's not something that is hidden. Rather, we all know the importance of drugs. And the drugs that we have been using is fine. We have been using a lot of drugs. There is nobody who doesn't use any kind of drugs. But the latest things that come, they are disruptive. We all heard about Ozempic. Ozempic was a disruptive drug. It changed the way we are looking at diabetes. We also have, I was looking at the drugs that India has manufactured. So I think there's a recent approval of nephromycin. This is an antibiotic that was recently approved from the Indian labs. But the situation is grim. The drugs that we are using, only 7 out of 900 approved drugs have been indigenously developed in India. This is after 75 years of our independence, that we have got only 7 drugs that have come to the market. So I think we have a lot of opportunity here in this particular field for doing something that can really disrupt. Disruptive means which is something going to change the life of the people. And drugs are life-saving. I can't emphasize enough to say that drugs are something that are going to change the humanity, and they are the ones that are also the economic engines of the country. It's not just about saving lives, but they are the economic engines. Billions and trillions of dollars are going to come from these pharma industry. So anybody in the policy who's thinking about revving up the engines of the economics, pharma is one of the things that should be done with emphasis. So we cannot do this alone. It's all about multidisciplinarity. And the drugs that were done earlier, that somebody has got a secret formula that is going to help some people, and then the formula goes away when the person dies, or it just carries on in a secret way. Now we know that it has to be carried on. The new drugs that have to come, they have to come from a multidisciplinary efforts. It's not just about engineering or biology. It has to be a mix of several disciplines. It has to be the expertise of the AI people now. It has to be the expertise of the chemists, the formulation scientists, the people who know the biology of the human body that is going to be affected by these drugs. So all these disciplines have to come together. But believe me, this needs a big, big change in the mindset, which also has to be disruptive. The mindset of working alone, mindset of doing things alone, mindset of having territories. No, this needs to be broken down. These silos need to be broken down. We need to come up for a cause. The cause is saving lives through the drugs. And I think apart from all the technical knowledge that you will get here, one thing that I would repeatedly say is to come out of your own silos and shake hands, collaborate for the cause. Right? So it's not my program. It's not the program of nanotechnology. It's a program for the university. It's a program for the country. It's a program for the humanity. We are talking here about, for the rest of 5 days, we'll be talking about drug discovery, how we can save lives. Saving one life is equal to saving the humanity. So thinking on those terms, thinking about the cause and rising up for the cause without any kind of hindrance is the need of the hour. This will also be a disruptive change in our mindset. So with that, I would also like to tell you about the Cancer Nanomedicine Consortium that we have made together using expertise from 7 different departments across the university and 3 different faculties. Again, this was something very new, that how these things can be done in such a way that people from different disciplines, different departments are coming together. But we have to know that this is the need of the hour, the collaboration, the expertise, not reinventing the wheel, rather talking about the collaborations complementing each other. And with that, I would like to introduce that we also have another course coming up in December about therapeutic targets. We have the foreign faculty coming from UK for that. And I would also like to thank, last but not the least, the vision of the people who have developed this course, GIAN, which is for the benefit of the Indian scientists, Indian faculty members, Indian students from the expertise that we have abroad, where we look forward to their expertise and bringing us to the knowledge that can give us a speedy output. So I would like to thank IIT Hyderabad and Ministry of Education, as well as the visionaries for the GIAN course, who have repeatedly funded and repeatedly organized these courses, which are very essential for us to go out and see the world through the eyes of our chief guests and the foreign faculty and the dignitaries that we have here. If we can't move abroad, at least we can see through their eyes. And with that, I would like to encourage all the participants to actively engage and actively participate in the 5-day course that we have here, learn and then disseminate. And one final message would be about collaboration. Again, once you are coming from different departments, different places, different cities, uh, different disciplines, talk to each other and develop this mindset of working together. Thank you so much. And I would like to, although there will be a vote of thanks later on, I would like to thank my small nano team, which has put this together. It's a small, very small effort, but I hope you'll appreciate that. Thank you.
Thank you so very much, sir, for delivering that very befitting welcome to this GIAN gathering. It is now my privilege to introduce the esteemed local coordinator for this GIAN program, Professor Vibha Sharma from the Department of English, Aligarh Muslim University. She serves as AMU's member in charge of public relations. As sir have already told, but I still would like to say it again, that she recently received National Awards to Teachers 2025 from the President of India. Her leadership as local GIAN coordinator has been pivotal to the success of our program today. I would now like to invite her to share her thoughts with us.
Thank you very much. A very good morning. Assalamu alaikum. Adab. I am doing the duty of Professor Jahangir Varsi, who's not here. I'm officiating as local coordinator today, so that should clarify the matters because his name is written in the card, so maybe it confuses the students and others who do not know about that. Since I have been associated with the GIAN coordinating in the past when he was not in university, so therefore this responsibility has been handed over to me. So I'm very happy to be here in a very different environment from mine because I belong to arts faculty. As you people know, I teach English and I've been teaching English for almost 25 years. The thing is that this particular course, which is sponsored by Global Initiative for Academic Networks, this is a program by Ministry of Education, and it is conducted nationally, coordinated nationally by IIT Hyderabad at the moment. In the past, it was with some other agency. Now, the process is that this program is applied for. There are applications we have to apply online, and when our applications are vetted, they are accepted, and then we are awarded this award, this particular grant, under which we are facilitated with invitation, with inviting a foreign faculty who stays at least for a week in India and interacts, conducts lectures, workshops, sessions, demos with researchers and participants. I think it's a beautiful concept. And as Dr. Aziz has already pointed out, that this is a wonderful thing that government has offered us with. I think we should never let it go underutilized as an opportunity. Let me tell you why I see it as an underutilization, because in my small stint, brief stint as the coordinator, I realized that there is a need for us to go beyond what is happening inside the room, inside the place where we are, because this is for networking, academic networking. That's something very important. And that's the reason I'm very happy because of the panel today here that includes, that represents academics, that represents industry, that represents scientists. So I think I should, clap for this conceptualization. It's a wonderful thing. First of all, I congratulate for the entire conceptualization. You should give a big round of applause to Dr. Aziz. Interdisciplinarity is nothing new in 21st century. Actually, now there is beyond disciplinarity, we are going anti-disciplinarity also. And because knowledge is a continuum and we can enter knowledge through any gateways, whether it's sciences, whether it's literature, whether it's humanities or social sciences. But if you look at any process that happens, whether in a science classroom or in humanities classroom, each process is interdisciplinary. And how? The fact that discovering drugs, any drug discovery, is not just a molecular discovery. It is not just a surprise that happens at the level of technology. It is a collaboration, as Dr. Aziz very beautifully put forth, that it is a collaboration with technology, science, computation, and I must also add ethics, because we see how when drug discoveries are celebrated and the euphoria that is around those drug discoveries, that sometimes overshadows the ethics behind it. And therefore, universities like ours are the best place to discuss these things, because a university is not just about one department. There are a number of departments wherein one classroom sciences are being discussed, molecules are being discussed, technologies are being discussed, then in the other classroom, ethics are being discussed. And when these minds come together, they join hands. They only work in the interest of the humanity. Any education that takes us away from the service of humanity is not education, because we don't produce consumers from central universities like ours. Aligarh Muslim University's motto is to produce people who serve humanity, who teach humanity, and who extend the mission of Sir Syed Ahmad Khan. So I think it's a wonderful project, and I'm very excited to see its result. In these 5 days, I'm sure the students, faculty members will have a great time brainstorming, interacting with the luminaries who are present here. I thank all of you for taking out time, for taking these pains to come to a small town like Aligarh, but I'm sure your presence has added to the greatness of this campus. And one more thing, being the local coordinator today, I must express that uh, we have completed, this is the 11th GIAN course of this season. We have one more to go, and that also is by Dr. Aziz. So Dr. Aziz is doing 2 courses this season. I have also done 1 course this season. It's been a very successful one, and I was also involved in one of the processes of one of the courses is conditioning when Dr. Aziz applied. So that also is a good experience, a gratifying experience for me. This year, we have applied for the complete capacity. That is, 40 proposals have been sent by our university. That was the maximum capacity for any university, and the process of reviewing has also begun. So let's hope that we will be awarded many more, and many more scientists, industry people, academics will be amongst us. We will be interacting in the name of academic networks and will be enriching the map of this university, that we always exist for betterment of humanity, nation, and everyone. Thank you very much. All the best for this wonderful course. Thank you.
Thank you so much, ma'am. I hope we make this program a great success. We are here to celebrate innovation and cross-disciplinary collaboration, and there is no one more fitting to address these themes than our next speaker, Professor Mohammad Muzammil, Principal, ZSCET, AMU. His dedication to harmonizing human capability with technological progress embodies the ethos of this event. May I now request Professor Muzammil to step forward and share his vision and wisdom with us all.
Thank you very much. Chief guest, Mr. Syed Ahmed. Guest of honor, Dr. Imadul Islam. Coordinator GIAN, Professor Vibha Sharma. Dr. Azhar Aziz, Dr. Ziauddin, faculty members, research scholars, and my dear young friends. A very good morning to all of you. Assalamu alaikum. Really a matter of great pleasure that I am here for this GIAN program. Very timely, very relevant in today's context. Earlier, we were not talking about multidisciplinary nature of activities in any domain of knowledge. It was specifically in one direction people were moving. Now the scenario has changed. Drug discovery and drug delivery, माजरत के साथ ज़ियाउर रहमान साहब से कि सिर्फ अब इनका डोमेन नहीं है. So pharma people cannot claim that this is going to be the domain of these people. Other people are also going to have a complete say in this regard. That is, a number of disciplines are going to act together to come out with new and better ways of human welfare. Because the challenges are different. Earlier, we were not talking about very deadly diseases like cancer, muscle dystrophy, neurodegenerative disorders, and many, many more. So every person is going to have different type of requirement, different type of treatment, and also optimization is one factor which is going to be there as far as different molecules and different drug deliveries are concerned. So when you are talking about optimization, engineers are going to play their role. When you are going to talk about different training of the subject, that what the past data is going to say, data analysts are going to come into picture. This recently, nanotechnology is going to take a big role in this direction. That is, at which point, at where the disease is going to occur, where we are going to put in those molecules so that minimum losses are going to be there and major benefit we are going to get there. Just see the example of mRNA recently. Chemists were there, biologists were there, material scientists were there, AI people were there, regulatory bodies were there. So this is a combination of all these factors when we had come out with this mRNA in a very short period of time, and that was a very challenging time, and people got benefited a lot. So this is our objective and this is our motto. That's why we all are here to come out, to think of, to get the knowledge of our learned speakers are here, that in what way we can have this amalgamated all these things and to come out this better ways. So I congratulate the organizers that they have organized this course, this GIAN course. And I'm sure you people are going to learn a lot with this course and going to use this knowledge to have something which is going to be better as far as humanity is concerned, human welfare is concerned. Some chips are being designed. We know that if a sugar patient is going to be there, that at a particular point of time, some insulin is going to be released and everything. This is all which is there, that is different ways of delivery is there. So we hope we are going to have a very successful this course and you people are enjoying it. Thank you very much. Thank you.
Thank you so much, Professor Muzammil. It is a profound honor to finally welcome Dr. Imadul Islam, our distinguished foreign faculty. Dr. Imad joins us from the King Abdullah International Medical Research Center in Riyadh, where he serves as a senior research scientist, deputy director of drug discovery, and head of pharmaceutical analysis. His career is a testament to global scientific leadership. Before his role in Riyadh, he directed CNS preclinical drug discovery at the Bristol Myers Squibb Biocon Research Center, Bangalore. His journey also includes foundational work as an investigator at GlaxoSmithKline in Singapore and a period of escalating responsibility at Berlitz Biosciences in California, where his research spanned the therapeutic areas of oncology, immunology, and cardiovascular diseases. The impact of Dr. Ahmad's work is both deep and broad. His early research on metabolic diseases at Synaptic Pharmaceutical led to a major collaboration. This vast industry experience is complemented by his academic roots as associate research scientist at institutions like Upjohn Pharmaceuticals. His career path is crowned by a formidable record of 32 issued US patents and 40 peer-reviewed publications. His current area of interest is antibody drug conjugates for treatment of cancer, their application in immunological and inflammatory disorders, and de novo design of new anti-cancer compounds. Dr. Islam, we are delighted to have you with us, and now we invite you to inspire us with your address.
Good morning and Assalamu alaikum, everybody. Coming to Aligarh is like a homecoming for me because my both parents, they studied long time ago here. And I see familiar faces, so I really feel very proud and happy. I would like to thank Dr. Azhar for inviting me to this course and Mr. Sayyid Ahmed, who I met first time as a chief guest, Professor Sharma, Dr. Zainuddin and Professor Muzammil. Thank you so much for being here and attending this course. It is my pleasure and honor to be part of this course, and I'm humbled by this invitation. I have been involved in drug discovery for a very long time, and almost Dr. Azhar and Dr. Muzammil has covered what I wanted to say, but inshallah I will try to say a few more things that working in pharma companies in almost 4 continents, I gained some experience, so I want to give it to our student here in next 5 days. Drug discovery is it is a multidisciplinary, as Dr. Azhar has put the title, it is a multidisciplinary science. And drug discovery scientist and as a practicing medicinal chemist, I want to emphasize chemist because I have some chemists in the audience. We are practicing medicinal chemist, and I think and I feel at the heart that the chemistry is the central science for drug discovery. I know Dr. Muzammil will not agree with me, but it's okay. It is a central science for the drug discovery. I would like to quote a beautiful word from 1987 Nobel laureate Jean-Marie Lejeune from Strasbourg. He said once, atoms are letters, molecules are words, and supramolecular entities like proteins are sentences and chapters. So it covers everything. And Dr. Sayyid, he was talking to me that he's going to talk about vaccine, and vaccines are also proteins. So I'm really interested to listen from you. It will not be complete if I don't recite a verse from Allama Iqbal, who says that Zindagi kya hai anasir mein zuhure tartib, maut kya hai ini ajra ka pareshan hona. Thank you so much.
Thank you, Dr. Islam. It is a pleasure to have you with us today. Dear mates, it is a great honor to welcome Mr. Syed Ahmed, the chief guest of this program and CEO of Tech Invention. Tech Invention is an international leader in affordable biotherapeutics and novel diagnostics. Under his guidance, it has become a certified deep tech pioneer and great place to work with 15 plus patents filed. In my opinion, he stands to be a visionary, and I use the term visionary deliberately for his dedication to a single powerful mission, making world-class healthcare a reality for all. He has worked at the helm of major vaccine business units at industry giants like Panacea Biotech and Abbott before establishing Tech Invention. He has led the development of vital vaccine and biologics across 30 countries from the heart of Africa to the communities of Latin America and across Asia. His leadership has enabled multiple global firsts, such as the development of first recombinant vaccine in Turkey. Sir, we are privileged to have you with us today. Please join me in welcoming a resounding welcome to give Mr. Syed Ahmed for his keynote address.
Thank you, ma'am, for the kind words. Thank you, Dr. Azhar, for having me here today. Dignitaries on the dais and off the dais, patrons and faculty, it's indeed an honor and a privilege to be here today. If there is one particular intervention that has impacted healthcare the most for over a century, it is vaccines. If there is one particular intervention which impacts the maximum sustainable development goals, it is vaccines. And today, the jargon, the buzzword is sustainability. If there is one particular intervention that impacts all aspects of sustainability, be it economic, be it social or environmental, it is vaccines. And adding to that, if there is one particular field where India has a significant market share worldwide as the world's #1 supplier, it is vaccines. I think all of us are really proud of that. But when you are right on top and you have a 50+ market share by volume of the world's vaccine supplies, the biggest challenge and task is to sustain and grow on that. And that, gentlemen, cannot happen without industry-academia partnerships. If you see the best of vaccines that came in, whether during COVID or prior to COVID, are all from academic institutions of research excellence. It is Oxford, it is UCL, it's the Cleveland Clinics, and for example, our own ICMR out here. And today, the objective of my visit to AMU is to discuss with the faculty and the patrons and students and come up with a concept for industry-academia collaboration where we could establish a vaccine consortium out here, which will focus not only on delivery, but also on design and discovery. That's the objective out here. We did establish a One Health Research Center in a center called Ganpat University in Mehsana about 1 year ago, and I must tell you, it's doing exceptionally well. There are cross-functional teams from the Faculty of Medicine, Faculty of Agricultural Sciences, and Faculty of Pharmacy, and very soon we should be doing very good translational work on infectious diseases. And when we are talking of vaccines today, vaccines are not just limited to communicable diseases, but also impact cancer. In fact, we have our first candidate, mRNA, for the non-small cell lung cancer. And we have a challenge when it comes to delivery, and that's where we look forward to Dr. Azhar's team to work out some cutting-edge concept on the delivery, especially around the lipid nanoparticle. Not just that, yesterday I was at IIT Bhubaneswar, India's first, or rather the world's first next-gen TB vaccine, which IIT Bhubaneswar has developed over the last about 10 years in collaboration with a Bharat Institute called ILS, was licensed to us yesterday, which is a matter of pride for a small company like ours. Week before last, ICMR gave us the technology for the next-gen malaria vaccine. And just about a week ago, the Department of Science and Technology Development Board has supported us for the next-gen pneumococcal conjugate vaccine. Armed with all these basic research candidates, I believe it is a fantastic opportunity for all of us in AMU to collaborate with the industry and come up with cutting-edge technologies which will help India not only sustain but grow on our current market share. Thank you.
Thank you so much, sir. May I now call on stage our Indian faculty, Dr. Mohammad Zainuddin. He is the Senior Director at Jubilant Therapeutics and has an expertise in drug metabolism and pharmacokinetics, which greatly enriches the scientific foundation of our program today. May I now please call him on stage?
Uh, yeah, thank you very much. Assalamu alaikum. And it's been a privilege to stand here and talk. And as the last speaker, I stand between you and your high tea probably. So I promise I will not waste much of a time. And usually it happens when you are a last speaker, then most of the time your stuffs have already been covered. So from the point, industry doesn't grow, as Ahmed Bhai told, that industry doesn't grow in a boardroom. It grows in a classroom. So this is the reason why I came back here and try to learn something new, and if that can help us to do it. So I will try to assess something more. There will be a lot of talk. I will trying to capture most of the stuff here. But more importantly will be the industry-academia partnership and how I remember Linus Pauling statement. He said that the best idea or a beautiful idea is lot of ideas. So let's have those ideas, feel free, talk together, punch out the questions, ask us all assumptions which we'll present you, throw your questions, we will try to punch back to you guys and have a good day, okay? So let's have a collaboration. So academia, industry is not the need, but it's a necessity now. And let's bring it to the reality. Thank you very much, I appreciate it.
Thank you so much, sir. I now invite Dr. Syed Ahmed Afzal to grace the stage and deliver the vote of thanks.
Respected Chief Guest, Guest of Honor, esteemed patrons, distinguished faculty members, and dear participants. A very good morning to all of you. Assalamu alaikum wa rahmatullah. As one of the organizers of the GIAN program, it is my privilege and honor to present the vote of thanks as we conclude the inaugural ceremony of our GIAN course on multidisciplinary approach towards drug discovery and delivery. First and foremost, I express our sincere gratitude to the Honorable Vice Chancellor, Professor Naima Khatoon, for kindly granting permission for this program, under whose patronage this event has been made possible. Our heartfelt thanks to our chief guest, Mr. Syed Ahmed, who is the CEO of Tech Invention, for his inspiring and insightful address. Our special thanks to Dr. Muhammad Zainuddin, Senior Director at Jubilant Therapeutics, for joining us today and for his continued support to this program through his valuable expertise in drug development. I also extend our deep appreciation to our guest of honor and foreign faculty, Dr. Imadul Islam, for joining us and for his contribution to this program. His global experience in drug discovery greatly enriches our academic environment. We are grateful to our respected patrons, Professor Nisar Ahmed, Dean, Faculty of Engineering and Technology, Professor Muhammad Muzammil, Principal, ZSCET, Professor Vibha Sharma, Local GIAN Coordinator, Professor Afsar Ahmed, Director, Interdisciplinary Nanotechnology Center, for their guidance, encouragement, and unwavering support. I would also like to highlight that Professor Afsar Ahmed, Director of INC, has recently been selected for the Lifetime Achievement Awards 2025 by Mycological Society of India in recognition of his outstanding scientific contribution. We are also grateful to the Registrar, Pro Vice Chancellor, the Finance Officer
for their administrative support and cooperation, which enabled the smooth organization of this GIAN program. I would also like to acknowledge the Government of India for launching and supporting the GIAN initiative, which has made programs like this possible across the country. We are especially grateful to the Ministry of Education for their vision, encouragement, and financial support. Their continued commitment to promoting international academic collaboration has played a crucial role in making this program possible. We are also thankful to the Account Section, General Section, Proctor Office, CAW, CEC, Land and Garden, and all other supporting departments whose assistance has contributed significantly to this program. We sincerely thank Professor Ziaur Rahman, Chairman, Department of Pharmacology, JNMCS, for graciously facilitating the tour and hands-on activity for our participant. A special acknowledgement goes to Dr. Muhammad Azhar Aziz, the course coordinator, whose dedication, planning, and leadership have been instrumental in organizing this GIAN program. His efforts have ensured that this event could take shape in best possible manner. Our sincere appreciation also goes to the reciter for the soulful Quranic recitation with which we began on this program. I would also like to thank Dr. Salih Naseem, who is also one of the organizers of this event. I extend my gratitude to our faculty colleague, Dr. Qasid Anwar, for his cooperation, and to our non-teaching staff, Mr. Ayub, Mr. Muhibillah, Mr. Asif, Mr. Abdul Rahman, for their hard work and coordination. I also like to thank Dr. Afsar and our PhD student volunteers, Ms. Zainab, Ms. Sabi, Ms. Swati, Mr. Aharas, and Ms. Suzanne for their valuable assistance. I also wish to acknowledge the core member of Cancer Nanomedicine Consortium for their cooperation and academic contribution to this program. My appreciation further extends to all faculty members, technical staff, administrative support, and student volunteers whose efforts have brought this program to this stage, including anyone whose name I may have unintentionally missed. And finally, to our participants who have joined us from various disciplines. Your enthusiasm and involvement are driving force behind this GIAN course. We look forward to your active participation throughout the 5 days of lectures, tutorials, and hand-on sessions. Thank you all once again for your time, your support, and your presence. Assalamu alaikum.
Thank you so much, sir. I again would like to share my sincere thanks for all the distinguished guests and all the faculty members who are present here today, and my dear mates as well. We will now conclude this inaugural ceremony with the AMU Tarana, followed by the national anthem.
विनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you so much, everyone. We will now break for high tea. Kindly reconvene by 12 p.m. in the same place and at the same room for the lecture sessions. Thank you. introduction. I just have to say that your lecture is on this campus. Okay, yeah, thank you. Welcome back, everyone. I hope you all had a pleasant break. We now begin with our lecture sessions for the day with Lecture 1, delivered by our chief guest, Mr. Syed Ahmed, on the topic Drug Discovery and Delivery Vaccines for One Health Perspective.
Thank you. When I was told by Dr. Azhar to deliver this talk, I asked him how much time. He said 45 minutes. Normally it takes a full day. So the challenge was what not to speak today in these 45 minutes. Nevertheless, I will try my best to cover to the extent possible. All of us are aware that One Health is a concept that is gaining ground all over. And simply put, One Health simply means we need to take care of human health, animal health, and the environment around us. And I'll try and my best to cover this in a very short, whatever 45 minute slot we have, but keeping the vaccine context in mind. See, this morning when I started off, I talked about how much difference can a vaccine make. But if I add a number to it, every single dollar that is spent on vaccines has a direct healthcare benefit of almost $16 and broader economic benefits of $44. हर एक डॉलर जितना आप इन्वेस्ट करोगे इतने benefits रहेंगे vaccines में. And one of the reasons why I'm repeating this, I will do it time and again in my today's interaction, inshallah, and the future interactions, is because I strongly believe we will be able to set this up out here. And as far as One Health is concerned, I just gave you a very brief perspective. All that we are saying is we cannot look at human health alone. Whenever we look at human health, we also have to look at animal health, and we also have to look at the environment around us, and it's possible. Now, Azhar Sahab told me to give a very brief overview of what I do and exactly kya karte ho. Okay, Tech Invention is a very new company. It's just 9 years into business. And how did I get interested in tech invention? I'm basically a pharmacist by qualification and background. And I was into all kinds of small molecules, therapeutics, which I enjoyed a lot. But way back in 2009, I was at the helm of affairs of one of the largest listed companies for vaccine manufacturing in India. I was heading the global strategy, global operations, and also heading the corporate strategy. And that was the time the first pandemic, as we know, as all of us know, stuck. Some of you were kids, I suppose, then. That was called swine flu. I'm not sure how many of you are aware. और उस टाइम पे there was a scramble for vaccines. इतने लोग दिन रात मुझे they started talking to me, reaching out from various parts of the world, trying to ये मुझे वैक्सीन मिलेगा कहाँ पे, and many countries started reaching out to me कि हमको वैक्सीन बनाना है, because very few countries in the world were manufacturing vaccines those days, and even to this day the same trend continues. So in 2009, since I was associated with one company, I could not tell them where to get vaccines beyond what I was doing. Okay? And I tried to find out any one company that can help a country establish a vaccine manufacturing infrastructure or ecosystem. But I could not find a single organization, a single consulting firm that could have the end-to-end bandwidth on how to advise a country, a region, to how to set up a vaccine manufacturing facility. So that was the opportunity that I saw at that point of time. And 2013 was when I was at the peak of my career. I was the youngest vice president at the age of about 32, 33. A very high-flying life, flying business class, all star hotels, US to Canada, Brazil to Singapore. I mean, having a very rich lifestyle. But then, the thought of setting up a company for vaccines which can offer end-to-end bandwidth kept haunting me. And in 2015 is when finally I came out and took the plunge and started a company
not in the name of tech invention, it was called Technovation. And at that point of time, I was very new to business because all through I was an employee. So I got partnered with a bigger group and I had a minority share and we started a company called Technovation, which did exceptionally well. एक साल में we did so well, we beat all expectations क्योंकि हमको projects मिले from Russia, from Latin America on vaccine manufacturing consulting projects. But then what happens is जब फेलियर प्रॉब्लम करता है तो सक्सेस ज़्यादा हो जाती है तो भी प्रॉब्लम करता है वी हैड सीरियस डिफरेंसेस ऑफ ओपिनियन बिटवीन द पार्टनर्स primarily around the ideological differences because when it comes to low middle income countries they cannot afford, so you will have to have tiered pricing. Many things, everything is not about profit, it's a lot about passion and purpose, and we had huge ideological differences to the extent that the company just fell apart. And when the company fell apart, I was literally thrown out of the company. Okay. So 2015, I was bankrupt. I had to sell my house. There was a team of about 12 people with me, and we did not have an office space either. And because my house was sold, I did not have even an address from where I could register the company. This was the situation exactly 9 years ago. From a very high-flying job, you come down to a place where you have gone below the ground. I had two options at that point of time: to go back to the industry, okay, because there were very high-flying jobs, and the second was pursue the passion with which you have come up. And fortunately for me, my family, my wife and my daughter, they said, "No, you should follow your passion, because later on, we don't want you to regret that there was this opportunity and you didn't take it up." And so we set up Tech Invention at that point of time. और एक चीज़ याद रखना, because you're all young, you're going to be budding entrepreneurs, you're going to be budding scholars, you're going to be budding professors, you will have troubled times. And when you feel that you've gone below the earth and you are buried, remember the Almighty has kept you there for a reason. It's not that you are buried, but you are planted. And that from there, when you sprout, your growth will be phenomenal. And when it is phenomenal, you come back more stronger, more wiser. And this is the story of Tech Invention in the last 9 years. From a stage of bankruptcy, today, we closed the books of accounts at about 40 crores revenue March, and we just got a valuation of about 250 crores. It is bootstrapped, jante ho na, which means we don't have any investors, we don't have any borrowings. We are cash positive from year 2. The reason I'm saying this is there are a lot of learnings. I was asking Dr. Vibha, is there a course on entrepreneurship as part of GIAN? Okay, because this is something with which I really want to inculcate and impart to students, budding entrepreneurs like you. And I want many of you to get into entrepreneurship. Not because I've gone through success, but I've had failures. And failure is a bigger teacher, a better teacher than success. Okay. Now these numbers are any investors. Okay, I mean, today, if I stand and talk to investors, most of them come back and say, what is your PAT, what is your EBITDA, and most of them are willing to invest. But the difference is, when I started off, I had to stay strong. I did not dilute. ये आप लोग को भी करना है. When you start off into any entity, any entrepreneurship, any aspect, don't dilute early. That was my first learning. आपको डट के रहना है, आगे जाना है, पेटेंट्स फाइल करना है, डिस्कवरी को टेक्नोलॉजी रेडीनेस लेवल से जहाँ तक लेके जा सकते लेके जाओ, and only then, when you feel that you are at a certain stage where you have to raise funds, only then you can raise funds. Today, in fact, the asset base of my company has also gone up systematically, consistently, year after year. Now, how did this happen? Now, this is again a second aspect which I wanted to communicate to all of you. जब हम कुछ भी initiate करने जा रहे हैं, because all of us, most of us will get into research, start having an ancillary revenue stream, either teaching or coaching or consultancy, which will start giving you revenues initially, and that's exactly what we did. We started with consulting and advisory. which is absolutely asset light. इसमें कुछ ज़्यादा आपको assets capital requirement की ज़रूरत नहीं है. We made money out of consultancy. We invested into licensing of vaccines, very good vaccines. And we invested in clinical trials, we invested in regulatory. And from licensing, we set up a state-of-the-art R&D center. And when we started the R&D center, मतलब एक से पैसे लिए दूसरे में डाले, दूसरे से पैसे लिए तीसरे में डाले. And when we started off again, we used the Government of India's incubation center at Faridabad, in THSTI, in RCB, we set up the first BSL-2 facility. And when we did that, although we were very small, we were open to training and teaching a lot of students. Faraz is here. Faraz, I can see you anywhere. I think Faraz was one of our interns out here, and he was interning out there as a fresher, I suppose. We also collaborated with DBT BIRAC for a postgraduate diploma program in biotechnology, which even today DBT BIRAC praises as very pragmatic center. And from the R&D center of at THSTI, 2 years ago, we have set up a real state-of-the-art R&D center and a pilot GMP facility in Navi Mumbai. It is called Horizon. And we do everything out there from bioinformatics to molecular biology to bioprocessing, formulation, end-to-end bandwidth. We have filed more than 15 plus patents, out of which 3 have been granted. And now is something that I want to really talk to the faculty out here. The regulatory stringency is changing year after year, month after year, month after month. पहले जैसा नहीं है. Even for preclinical batches of a protein-based product, you need a GMP-like facility. You will have to apply through Form CT10. Inspection के लिए लोग आएंगे आपके पास, और कौन सा inspection? Joint inspection होगा. State और center का FDA. They will come, they will see what kind of infrastructure you have. Do you have the unilateral flows? How do you handle your biomedical waste? What kind of SOPs, what kind of training, and only then they will give you permission to take batches for preclinical trials, animal studies. Okay. So when we start doing any research work as a research scholar, we'll start looking at it from a translational perspective, और ऐसा होना नहीं चाहिए that you reach a certain stage and there is a valley of death. Look at it from an end-to-end perspective. BIRAC का एक grant है, Biotech Ignition Grant. आप लोग जानते होंगे. This is the only grant which gives about 50 lakh rupees at the ideation stage. And it is the most competitive grant, TRL 1 से TRL 3 तक. अच्छा, अब अगर इसमें जब आप qualify हो जाओगे, in fact we were one of the grantees, तो आपको TRL 1 से TRL 3 का ideation के ऊपर सवाल तो होगा ही, but more importantly they will ask you how will you take it to market. मतलब आपको उस तरीके का एक vision दूरंदेशी चाहिए. So even at a very, very basic research stage. And until unless that happens, the entire process of your research will come to a standstill. Now from there, now we are getting into setting up a commercial scale vaccine manufacturing facility in Mumbai, which I will talk about in some time. Consultancy mein today we are advisors to Asian Development Bank, advisors to World Bank, United Nations, operations of Project Office. In सब के बड़े बड़े vaccine infrastructure projects हैं where we are advising, and alhamdulillah we are doing quite well. When it comes to licensing in, we have done some very good licensing. The world's first recombinant pertussis vaccine, we did the clinical trial and it's due for approval. We have varicella, hepatitis A, all these vaccines is something that we've licensed in at very late stage. And today, our R&D center is all built around sustainability. I would encourage all of you, at least the faculty out here, to send out batches to our R&D center. Okay. We will be happy to train, coach, provide internship for about a short term, maybe about a week or so, followed by a more intensive program. Yeah, but then the challenge is we have to accept that Mumbai is an expensive place. So we will have to find ways and means on how to manage the resources together. Okay. And here everything is around sustainability. Even the air handling units that we use consumes 40% of the actual electricity. We have what is called as a zero liquid discharge. कोई भी इस waste में नहीं जाता है. After we inactivate the liquid discharge using the kill tank system, it all gets evaporated using these ZLDs. These are the contemporary concepts that are required from a sustainability perspective. And this is the R&D center that I was talking of. It is fully functional, and I would like to have you all visit. And when it comes to our indigenous portfolio, this morning I talked to you about 2 or 3 vaccines where we have taken the technology from outside, but inshallah we will be coming up with the world's first hexavalent vaccine, 6-in-1 vaccine for meningitis. Pfizer के पास 5-in-1 है, GSK के पास 5-in-1 है, Serum Institute के पास 5-in-1 है. We are getting into 6-in-1. और अगर आपने ये 6 serotypes meningitis के club कर दिए, you are covering 100% of the pathogens that are responsible for meningococcal disease. And this is a very, very important vaccine for Hajj and Umrah. It is one of the mandatory vaccines, ACYWX. But if you combine it with B, because B is part of global north prevalence and ACYWX are prevalent all across. And when Dr. Saab talked to me about visiting Saudi Arabia, I'm coming to you, sir, with this, because if Saudi Arabia can take it up, I think we will have a fantastic product coming out from India, which can benefit all, because that's one place where 150 countries, millions of pilgrims come together, and we need to protect them. And I think this is what I will share with you. And if this comes through, Azhar Sab, this visit would be the trigger and initiation for that. Okay? And there are many more such cutting edge technologies that we are working on. और इसमें क्या है? It is not just about discovery. Delivery is much more important. When we are talking of delivery, there are 2 things that we look at when it comes to vaccines. One is delivery from a formulation perspective. The second is delivery of reaching it to the most required vulnerable populations which reside in the rural part. Almost all vaccines are thermolabile. They need cold chain. तो हम लोग उसको last mile delivery की बात करते हैं. Okay. So when you're talking of delivery, it's not just the formulation on how to administer, but it's also how to make sure it is accessible and reaches the desired point of care, point of use. So delivery from both the perspectives are very important from a vaccine context. And this is what I want to talk to you about in some time. Now, this is the technology we got from ICMR for the next gen malaria vaccine. We are one of the very few companies. When ICMR, we were pleasantly surprised last month, called up and said, you are one of the recipients for our technology. And we were so happy when they said, हम इसका कोई पैसा नहीं ले रहे हैं आपसे. 0 milestones. If the product is successful, you just pay 2% royalty. That is the kind of confidence and trust that organizations like ICMR will lay on you, provided you do good groundwork, when your fundamentals are strong. And this was what happened yesterday this was a press conference at IIT Bhubaneswar and ILS, where we in-licensed the next generation of TB vaccine. I'll talk to you a little more about it when we go. When we were getting into it, we were spotted by DBT BIRAC, and this is what happens when we work very closely with the academia. We worked very closely with THSTI, and I was very, very pleasantly surprised when we were invited to present virtually to the Prime Minister of India on the concept of nudging the DNA with a handful of startups. Okay, this is the kind of visibility that you get when you partner with the academia and industry. And from there on, there was no looking back. We have got so many accolades. Okay. When we get into entrepreneurship and when we get into setting up a business, it is not just about profits. We will have to look at it from various perspectives. One of the most important aspects, even for an institution, an organization, academia, is good corporate governance. The world's #1 rating agency is Dun & Bradstreet. And last week, Dun & Bradstreet upgraded us from a low-risk SME to a lowest-risk SME. Okay, which means that there is nothing beyond this in terms of corporate governance, and they look at it from the point of business strength, liquidity, profitability, payment behavior, sector performance, and all that. And in addition to that, we just converted from private limited to limited. From an entrepreneurship perspective, when you start looking at a startup, आप शुरू में क्या करते हो, proprietorship firm set up करते हो, because it's easy to set it up. उसके बाद LLP बनाते हो, उसके बाद private limited में जाते हो, उसके बाद limited में जाते हो. Limited is the highest level. But let me tell you, if you are very, very serious about putting up something, not only for yourself, but from a legacy perspective, don't start with proprietorship or LLP. बायरैक डीबीटी चाहे डीओपी के आधे ग्रांट्स का क्वालिफाइंग क्राइटेरिया प्राइवेट लिमिटेड कंपनी के ऊपर होता है, विच मीन्स यू आर सीरियस अबाउट इट. द कॉम्प्लायंसेस बहुत ज़्यादा रहते हैं इसमें, बट इट इज़ वर्थ इट इफ़ यू आर नॉट थिंकिंग लॉन्ग टर्म एंड इफ यू आर लुकिंग अबाउट सीरियस बिजनेस. Last year, we were part of the Forbes India 200 listing, and I think we are one of the only companies into serious biotech which got into this. And the most important thing for a company to sustain and grow, when you saw those numbers, is not just the strategy, it is the culture that matters. Culture, the way you treat your employees, the way the camaraderie exists. और इसमें world का number 1 organization है, it's called GPTW, Great Place to Work. Okay, Great Place to Work, they rated our organization 2 consecutive years, and both the years we have beaten the industry in all parameters of credibility, respect, fairness, pride, and camaraderie. So when you have a strong fundamentals, it's not just about strategy, it is about culture, because it is people who will deliver the numbers for you. And using those numbers, you will invest and generate into value-added products. Going forward, the quality aspects are very important. ISO certifications is what we got. DSIR recognitions is what we got. Publications are important, but more important than publications are patents. Keep in mind, please do not publish a paper unless you have put up a provisional patent application. उसके बाद आपको 1 साल का टाइम है to go ahead and file the full application. हम लोग क्या करते हैं, when we have come up with a nice concept as a research scholar, we are very eager to publish, which is a part of academia requirement. We respect that. But if you feel that there is a translational possibility in this, discuss with your professor, discuss with your guide, file for a provisional application, and then publish it. Very important from a long-term perspective. And then, of course, I'm not going to talk much about IP here for want of time. You do it through the PCT route, not just the national phase filing. Because today, we have to look at it from the world, not just India. Okay? And it's not very expensive. And when you set up a company, like a startup, the DBT BIRAC system, they pay for your patents, they pay for your filings. Let's utilize those. Why can't we have a small incubation center out here, which is very, very specific, which is sector agnostic, and you will have all the privileges that the government infrastructure and ecosystem enable. And we will be very happy to support and collaborate with you on that. Okay, so let's look at it from that perspective. Now, coming to this aspect of vaccines from a One Health perspective. 70% of the total volume of vaccines procured by the UN healthcare agencies goes from India. Okay, all of us are happy about it, and this morning also we said we take pride in this. But what kind of vaccines do we supply? This is more important. We supply very, very affordable EPI basic vaccines, 5 rupees, 10 rupees wale vaccines. Okay, and we supply only from the private sector, 5 companies. 5 companies account for 70% of the UN procurement from India. I was in Bali last month by ADB PATH meeting, and I was asked to speak on Indian vaccine ecosystem. I told them about 10 companies active, 5 supply. Immediately after me, my Chinese counterpart came in and said, I have 75 companies, out of which 50 are active. Okay. Which means, are we going to sustain, and how are we going to sustain? Especially when we are looking at the low-cost vaccines. And whenever we launch low-cost vaccines, multinationals exit from there. You will not find a GSK, Sanofi, Merck Dome supplying TT, TD, BCG, hepatitis B. उनको उसमें interest भी नहीं है. they graduate and move to the next generation of high value vaccines. And that is where we need to focus on academia for basic research. Identify those lead candidates, identify the delivery mechanisms, and start working towards it in collaboration with the industry. And that's most important. I'm not touching on the third point, which is a regulatory requirement, because that's a session by itself. That's called the National Regulatory Requirement of Maturity Levels. Now, it's not going to be easy. During COVID, there were at least 5 large established pharma companies who came into vaccines. Today, none of them exist. They have gone back to doing what they best know, pharmaceuticals, which means it's not easy. During COVID, there were 50 plus startups who came into vaccines. Okay, today, 2 of them survive, one in Bangalore, one of them in Hyderabad. Most of them have either been acquired or they have wound up. What does it mean? I'm not trying to scare. But when you move your cards well, go about it with a very systematic, strategic plan and intent, it is possible. And when you do that, you are in a niche. Nobody can touch you. You cannot play the generic game here out here in vaccines. And this is an advantage for us today in India, because it's just handful of 5-10 companies whom you're going to compete with, unlike what's happening in our neighboring countries. You have heard of CDMOs and CRDMOs, contract research and contract drug manufacturing centers. There are 26 of them who do large molecule biotech. Not one does vaccines end-to-end in India. That's a huge opportunity. And that's where, again, if you're talking of CDMO, it's not contract drug manufacturing, it's CRDMO. And when you have an academic collaboration, the West looks at you more seriously. And especially for a prestigious institution like AMU, because this is how the ecosystem operates in the West. And that's how they've been able to turn out cutting edge technologies and cutting out molecules. Today, we say we have 70% of the world's volume by UN procurement agencies, but how many companies in India manufacture mRNA vaccines? Just one. Just one. And that to the conventional. How many companies in India have the adenoviral vector technology? 0. How many of us manufacture OMV-based platform technologies? 0. And these are the technologies that are technologies of the future. So if you're looking at doing any kind of research, let's look at contemporary platforms, not the conventional platforms. And there are so many vaccines which are high priority, which are not manufactured in India. So on one hand, we are talking of global leadership. On the second hand, there's a huge gap. And what are we sitting on? We are sitting on low cost pile of huge volume. Okay? And that's where we need to bridge. And there are opportunities out here. And if there is any one field that is the best funded for research, it is vaccines. Okay? You have the Gates Foundation, you have the Wellcome Trust, you have Carbex, you have GARDP, you have innumerable organizations we're going to fund. I'm skipping some of these points for want of time, but also the fact remains that when I decide which vaccine to choose from, okay, one of the key criteria that I will choose is the pre-qualification criteria of WHO. WHO has a criteria called pre-qualification, which means if my vaccine gets pre-qualified, I have a very high potential or propensity to take part in the global tenders and bids of UN healthcare agencies. And this changes depending on the demand-supply gap. And this is where we need to focus. There are high priority for pre-qualification, low priority for pre-qualification, no priority for pre-qualification. So if I'm working on a lead candidate or a target, I will not work on low priority or no priority. It may sound very interesting from an academic perspective, but it will not have any value when it comes to the commercial aspect, because there is no demand supply gap. There are adequate manufacturers. And this list is a dynamic list. It is in the public domain, and you can easily see it. You can easily see how from 2018 to 20 to 2024, 26, the priority changes. And so what we were talking of, the meningitis vaccine is a high priority for pre-qualification, at least the combination vaccines. And another aspect out here, and when it comes to vaccines, is combination vaccines help because it reduces the number of pricks, the number of visits to the clinic, the number of cold chain requirements, and that's where formulation comes in very, very, very handy. Okay. You use novel adjuvants, you use stabilizers, and the formulation department, especially if you have a nanotechnology-based focus, it's all the more better. Okay. And this is the medium priority, and you have a no priority and a low priority. And for the first time, to enable researchers and academic institutions, for the first time, WHO last year has come up with vaccines where the focus should be on research. And what does it have? COVID, of course, now no, but novel OPV 134, monkeypox, tuberculosis, Shigella, Gurubupi, Streptococcus, and Sabin IPV 19 S strain, which means the global organizations are telling us there is a huge demand supply gap, and if you are going to focus, if you want to make a success of it, focus on these candidates. And it's all there in the public domain. Okay. And the second aspect that we need to look at is antimicrobial resistance. All of us are aware if there is one aspect that is causing more deaths than accidents and cancer put together, it is antimicrobial resistance. Simply put, it is resistance to antibiotics which is creating havoc all across, happening not only in humans but also in animals. Okay. Now out here, there is a bacterial priority pathogen list which WHO has put up in 2024. You have a critical group, you have a high group, high focus group, and you have a medium group. The pathogens are clearly listed there, and most of these don't have vaccines. You take a Pseudomonas aeruginosa, you take an E. coli which causes the urinary tract infection, you cause A. baumannii, these are all nosocomial infections in hospitals and so on. None of them have a vaccine. These are opportunities for us. And when you do this, there are specific bodies in AMR which will fund you. There are some which fund you from TRL 1 to 3, some which fund you from TRL 3 to 6. And so you can actually plan and map out whom to work with. There are specific organizations like the Rights Foundation. If you collaborate with a university in Korea for this, there are specific grants that will keep coming to you. And such grants, bilateral country trade-related grants, are much more easier to access. In fact, we are in advanced stage of securing one for in vitro diagnostic for point of care tuberculosis. So let's look at it from the bacterial pathogen priority list. Trust me, none of them have any vaccine. And they are a dime a dozen. So when we look at vaccines, we look at it from seasonal limitations, which are seasonal diseases. There are residual unmet needs. In fact, there is a long list of unmet needs, the related vaccines, persistent global threats, potential outbreaks, neglected diseases, and nosocomial challenges. I'm not getting into each of these for want of time, but yes, I'm leaving the copy of this presentation with the organizers here for circulation. And at any given point of time, if you want anything further on this, if I'm not available, my team will be very glad to help and support you on this. Now, we talked about human vaccines. What about animal vaccines? For every 2 cattle vaccinated, 1 person avoids hunger. A study is published. In fact, I would say animal vaccines are as important or even more important than human vaccines. Every outbreak that we have seen is of zoonotic origin, मतलब जानवर से आदमी में आया है. आप Zika ले लो, आप Ebola ले लो, आप COVID ले लो, आप swine flu ले लो. How will you break the transmission? You will have to take care of the animals as well. And these zoonotic diseases are of very, very high importance. And not just that, when you look at an agrarian economy like India and also most of Africa, some of the South Asian countries, a lot depends on livestock. Now comes the interesting part. This is more for researchers. ये platform technologies हैं जो humans में use हो रहे हैं. Okay? There are inactivated vaccines, subunit vaccines, mRNA vaccines, and so on. Now, if you look at animal vaccines, there is a 100% overlap on the platform technology. जो यहाँ पे यूज़ होता है वहाँ पर भी सेम प्लेटफॉर्म यूज टेक्नोलॉजी ही यूज़ होता है. ओनली थिंग इज इन एनिमल्स थोड़ा कॉम्प्लेक्स है क्योंकि वहाँ पे वाइल्ड एनिमल्स अलग हैं, कंपेनियन एनिमल अलग हैं जैसे कि पेट्स हैं घर पे, एंड लाइफ स्टॉक. वी फोकस मोर ऑन लाइफ स्टॉक बिकॉज दैट हैज़ द मैक्सिमम इम्पैक्ट. But the same technologies apply here and there. So when you pick up, yesterday when I was in IIT Bhubaneswar, I said, when you're doing the TB vaccine, 2030 tak TB Mukt Abhiyan, Pradhan Mantri is on. Okay, and you need a vaccine for that. But jab tak aap human tuberculosis eradicate karte chale jaoge, bovine tuberculosis, जो गाय भैंस में हो रहा है वो बढ़ रहा है, और इंडिया में वही स्ट्रेन यहाँ पे है जो वहाँ पे भी कॉज कर रहा है. Until and unless you eliminate that, how will you eliminate tuberculosis? Malaria, okay, caused in humans through vectors, mosquito-borne disease. There is a new type of malaria which is coming up all across South Asia. आप कभी Google करके देखना. It's called monkey malaria. It has already got resistance to human drugs. Okay, and it is coming in from the monkeys into humans. Okay. In fact, elephants in Kerala recently have been spotted with chikungunya. I mean, you can imagine what's happening out there. And this is an opportunity for life science researchers, opportunity for even students in the engineering side, because like we talked about this morning, it's a cross-functionality, interdisciplinary aspects which come together to create cutting-edge technologies. And that's when, if you look at the total value and volume of the markets, human vaccines का fraction है animal vaccines, whereas the animal का population ज़्यादा है. Human में grants बहुत ज़्यादा हैं. Okay, अब you just see the kind of number of patents that have been filed. Okay, the red ones indicate for a particular platform technology in vaccines. The red one indicates patents filed in the human side, and the blue indicates patents filed for the animal side. When it is the same platform technology, okay, when I am exploring a lead candidate, when I'm trying it on humans, why not I also customize it to suit the animal ecosystem and try to file a separate patent out there, develop a separate formulation out there. It helps. Okay, I'll give you another challenge, and this is to Dr. Azhar's team. सब वैक्सीन दिया जाता है 0.5 ml. Okay, most of it is given through the intramuscular route. The same vaccine अगर आप इंट्राडर्मल देते हो 15 डिग्री केस में 1/5 हो जाता है डोज. From 0.5 it comes down to 0.1 ml. Okay, the challenge is how do the healthcare workers administer it intradermally? This is called fractional dose, and this is proven and it is allowed by WHO, government of India. जब rabies vaccine का shortage आया हर जगह पे, so the government started advocating it by ID route, 1/5 the dose. मतलब अगर 500 रुपए का है तो 100 रुपए हो गया. और जब आप एक को इम्यूनाइज करते हो इंट्रामस्कुलर रूट से, आप वही एंटीजन को उतने ही क्वांटिटी में 5 लोग को इम्यूनाइज कर सकते हो. एंड इंडिया इज एन एग्जाम्पलरी एग्जाम्पल. वी इम्यूनाइज ऑलमोस्ट 20 million babies every year with ID route of IPV, but there is no device which is a low cost device. एक low cost device चाहिए जो आप यहाँ पे रखे और वो intradermally चला गया. And that this should be an engineering marvel. We need a low cost device out there. And I'm telling you, if we are successful with this, we have some prototypes and we are looking for partnerships out here. We need an engineering faculty, we need medical faculty out here, and we need, of course, formulation experts. If we come together and develop, believe me, it's $1 billion product. Human papilloma virus का जो vaccine है, it is proven 1/5 the dose ID route. Influenza vaccine, it is proven 1/5 the dose ID route. Injection इसमें injectors हैं, auto injectors already are available. I'm telling you that. Novo Nordisk के पास है, PharmaJet है, all US based, but very expensive, beyond the reach of a common man. And that's where innovation comes in. And that's where institutions like AMU should start working on. And this is something that we can do. And this works not only in humans but also in animals. Just that animals में थोड़ा device अलग करना पड़ेगा क्योंकि animal का hide अलग रहता है. उनका dermis का और subcutaneous के जो angles रहते हैं वो अलग रहते हैं. So you need to customize it. But there is huge opportunity. And if we can strike this balance and make it equal between humans and animals, then you are leveraging your technology to a broader subset of people. Okay. Let's look at it the last 5 minutes. India is #1 in livestock population worldwide. We are not only #1 in human population, we are #1 in livestock population also, which means more the animals, more the disease. Okay. Achha, we are #3 in egg production, #1 in milk production, #1 in buffalo meat production, #2 in goat meat production. And how much losses do we have because of vaccine preventable diseases? At $7 billion every year. तो यहाँ पे opportunity बहुत ज़्यादा है to focus on. And remember, it is the same platform technologies that are used. और यहाँ पे एक बहुत अलग सा challenge है, and this is again something very interesting for the medical faculty out here. Human में पता चल जाता है कि अगर मेरे पास एंटीबॉडी है वो वैक्सीन से आया है या डिसीज से आया है. Because I know I have been immunized, I can talk. Animal livestock में कैसे पता चलता है? It is because of the vaccine or because of infection. That is why the World Organization for Animal Health in the last 5 years is looking at what is called as a biomarker in every vaccine called DIVA. DIVA बोलते हैं इसको. Differentiating between immunized and vaccinated animals. You need a biomarker which will prove अगर antibodies दिख रहे हैं किसी animal में, is it because of infection or immunization? Then I can separate out that particular flock, that particular animal, prevent it from infecting the herd and protecting the farmer's life. This is again a huge opportunity. Except for foot and mouth disease in India, no vaccine has a DIVA biomarker. And this is something that we can definitely work on. और यहाँ पर हम सब लोग कार्बन की बात करते हैं, कार्बन एमिशंस, which is the next most dangerous greenhouse gas, मीथेन. और सबसे ज़्यादा मीथेन कहाँ से आता है? एग्रीकल्चर से आता है, लाइव स्टॉक से आता है. लाइव स्टॉक से फर्मेंट करके ना गाय भैंस, they emit methane, and this is a greenhouse gas. And it is proven with studies, innumerable studies which I will show in my slides, where जब एनिमल को डिसीज होता है तो मीथेन एमिशन बढ़ जाता है. जब आप उसको वैक्सीन देते हो, डिसीज प्रिवेंट होता है तो मीथेन एमिशन कम होता है. आप सस्टेनेबिलिटी पे काम कर रहे हो कि नहीं कर रहे हो? जब आप एक फ्लॉक को इम्यूनाइज करते हो तो देयर हैव बीन अ वेरी बिग स्टडी इन नाइजीरिया which has pulled out 2 villages out of extreme poverty after animals in those villages were immunized, the livestock animals. Which means what we are talking about here is sustainability from all aspects. 70% of rural India depend on agriculture and livestock for their income, and 70% of the households who tend to these animals are women. तो जब आप animals को immunize करते हो तो आप women को empower कर रहे हो, because healthy women, healthy animals, and they are being taken care of by women who tend to contribute to the household income through the livestock and cattle. This is how rural works. छोटी piece of land, थोड़े गाय भैंस बकरी, थोड़ा poultry, and that's how the agrarian economy works. And that's where immunization is very important, and it impacts all aspects. And I'm skipping these slides because I'm running out of time, but these are all the studies that have proven extensively and beyond doubt. And the government of India is highly supportive of any intervention you do in the livestock segment. The Department of Animal Husbandry and Dairying has become a separate ministry by itself only to focus on the animal health. And there are specific grants out here which we can look at. So there is a stark difference between human and veterinary ecosystems, and in every aspect there is a difference, and we can definitely make a difference for that. I'm just skipping some of these slides because I just have about 4 or 5 minutes left. But yes, in conclusion, what are the aspects that we can work on at AMU? First is if we can come up with something which can contribute to thermostability. Cold chain hatana hai. Room temperature पे supply करना है vaccine को. There are no room temperature vaccines anywhere in the world. It is possible. बहुत बड़ा काम होगा इसमें. Billions of dollars will be saved. Thermostable vaccines is a big thing, and there are huge amount of grants for that. Second, can we develop some novel adjuvants from a chemistry perspective? Most of the adjuvants are chemical moieties. We have alum-based adjuvants, squalene-based adjuvants. Adjuvant क्या करता है? Antigen का quantity कम कर देता है. आप adjuvant डालते हो तो antigen का requirement कम हो जाता है. We need antigen sparing action. Can we work on novel adjuvants out there? Third is, can we come up with affordable devices which can reduce the dose from 0.5 ml to 0.1 ml? And fourth is, can we focus on intellectual property and push-pull factors with all these aspects together? ये 3-4 simple चीजें हैं, because there are too many things if we get into, then we will not be successful. What is doable, what is possible, and where there is potential. Now, to enable this to happen, we are setting up a manufacturing facility in Mumbai. It's called the Global Collaborative Center for Medical Countermeasures. We already have got into MOUs with VaxHub, which is Oxford and UCL, Bactivac. Very soon we'll be signing up with Cleveland Clinics. हर जगह funding का crunch आ गया है, you know, after the regime change in the US. And people want to offload research to places who do good research, quality research, and affordable research. And our objective out here in this center is to bring in candidates from academia, research institutions, and take it through the complete translational route all through the way up to marketing authorization and WHO pre-qualification. This is a very unique model that we are working on, and this model has got the government support. Last week, you can see the date there, 21st of November. This is a press release from DST and TDB. The government has said. Okay. And this is the current status of the project on the left-hand side. Inshallah, in about 2 quarters from now, we will complete the project. And any of you have any thoughts, we will be happy to collaborate, bring it down here. And this project at the World Local Production Forum in Netherlands, Hague, was rated as the #1 project in terms of impact across low middle income countries. And this is where I would like to collaborate with AMU and wish you all the best. Thank you.
Thank you so much, sir. We will now take the next 15 minutes for an open discussion based on the lecture delivered. Participants are requested to keep their questions concise and relevant.
No, I think vaccines is a new subject. So it takes time. I quite understand. Yeah. Yeah. Okay. The first thing is vaccine is a protein. It's a large molecule. Okay. You can come up with novel adjuvants using combinatorial chemistry, the concept and the context. And that's a very, very important point, one. Second is जो हम उसमें stabilizers use कर रहे हैं to make it thermostable. You can apply it there as well. Okay. So both these aspects that I already talked to you about, which makes it thermostable and gives antigen sparing action, can come in very handy. So it's not purely about biology and protein, large molecules. Small molecule chemistry will come in very handy out here going forward. Therapeutic. No, I think, sir, you've raised a very, very valid and a pertinent point. Ever since the CGT concept has taken off, cell and gene therapy, especially around the CAR-T and so on. So people at, especially in the center, I'm talking of India, have come up with specific grants around that. But about 6 months ago, I had a personal meeting with Dr. Rajesh Gokhale, who is the DBT secretary. And we made a very short presentation to tell him what Moderna, okay, and BioNTech, just two companies as case studies, are doing. Moderna and BioNTech have 3 candidates each in the arena of therapeutic cancer, and all of them in advanced stage of clinical trials, phase 2, phase 3, and so on. And then we also gave an examples of 1 or 2 Korean companies. And we said in India, hardly anyone is working on it. Sequel to which, they came up with a first grant on mRNA-based biotherapeutics. This grant just got released last month, which means the awareness levels we will have to create. And if you're able to, today at least the government is quite open, what I can mention. You put up an application, ask for an appointment, meet Dr. Jitendra Kumar, the CEO of BIRAC, meet Dr. Rajesh Gokhale, meet Dr. Jitendra Singh and DST, and come up with a very short presentation. And I'm sure they are all yours. So the awareness levels have already gone in, and every time we will have to show a little bit of fear कि देखो वहाँ पे इतना सब हो रहा है हम क्या कर रहे हैं, and that's when they get activated. तो उसके लिए काम शुरू हो गया है already. One. Second is, I think we need to combine or collaborate with leading cancer research institutions, okay, especially organizations like ACTRAC, or for that matter AIMS at Delhi. Now these organizations have a very positive perception in terms of research when it comes to the authorities. When we get into some kind of an association with them and put up a joint application or a joint appeal, it is heard much better. Last but not the least, when these things happen, one of the biggest challenges are, if there is no regulatory pathway, then how do I go about conducting my research? That's the biggest challenge. That's when we need to also involve the regulator and appraise the regulator, because unless there is a regulatory guideline, आज तो research करने के लिए भी regulatory guideline है beyond TRL3. So ये 3 लोग को हमको appraise करना है, and there are ways of doing it. They have opened up 2-3 also hotlines. One is called e-Vartalap, which the regulator has opened up. You can put it up online and the window, and they respond in 48 hours normally. DBT has also has started a concept called Patent Mitra. I'm sure some of us are aware of it. Usme you can put it up if you have a new concept, new idea, and they will definitely come back to you with people from different authorities, and they will tell you different regulatory bodies, different authorities who will come together and support you on how to take this ahead from a translational perspective. So all these things are happening, just that we need to put some concentrated effort and it will happen. Yes, sir.
Thank you for enlightening us about the vaccines. Although it is not my area, not my core area, I will call, but we are doing some work in the vaccine area. And, you know, from Saudi Arabia recently, there was a Merck's vaccine. And it comes and goes, comes and goes. And clinical trial is very difficult to do in vaccine. So can you shed some light on, you know, in SARS, we rushed the clinical trial and we have the side effect. I don't know whether it is true or not. Although I have taken all 4 vaccines from Moderna and Pfizer, I don't feel anything, but people say that. I don't know how to believe it or don't believe it. But can you talk about a little bit of clinical trials of your vaccine and how you're going to overcome that?
Yeah.
Because in drug discovery, it is 10 years almost.
Yeah. So for vaccines which are already listed by WHO as priority for pre-qualification, there are very clear pathways. There is something called as QTTP, Quality Target Product Profile. Okay, and if you follow that, then we don't go astray. Then in the clinical programs, you will not have challenges with the ethics committees and so on if you follow that one. Second is, if you're talking of a novel vaccine, okay, supposing I'm coming up with a vaccine which is a second or a third. Like in case of pharma, you have bioequivalence and bioavailability studies where you prove. In case of vaccines, we do what is called as a non-inferiority study. Okay, you take a licensed and a registered vaccine, and I need not prove efficacy with my vaccine. I only have to prove that my vaccine is non-inferior to the innovator or any licensed vaccine from an immunogenicity and safety perspective. That becomes less cumbersome. Third, there are diseases which have very small pockets and outbreaks. Some of them are very serious, like you have Lassa, you have monkeypox, you talked about MERS. उसके लिए there is a separate organization और इसमें ना कोई पैसे नहीं डालता because मैं बना भी लूँ तो आउटब्रेक आएगा तो कोई 10,000 डोज लेगा. It doesn't make business sense. So there is an organization called CEPI, CEPI, Coalition of Epidemics Preparedness Initiative. Okay, if you Google CEPI, you will come down. CEPI has listed down such pathogens which are rare and have periodic outbursts here and there, and they know it is not profitable for the industry to invest, so they invest. And MERS is a priority pathogen for CEPI. Now coming to MERS, the challenge that we have had discussions with the authorities in GCC is, for a long time we thought it is coming from camels. And we said, whom do we immunize? Even if you come up with a good antibody against MERS, do you need to immunize humans or do you need to immunize camels? Or do we need to break it? But fortunately, over the last 3 years, there have been no outbreaks of MERS. Almost the last outbreak was just prior to COVID, if you see. So after that, the interest in MERS has gone down, but 2 companies are being heavily funded by CEPI to work on a vaccine for that, and which is already in phase 1. One is called Bavarian Nordic in Europe, so they are working on it. And the second is another company, I'm not getting the name, but they are working on an mRNA vaccine for MERS. So possible. But again, coming back, we are not today very sure that the transmission is happening from camels after all these years of publications and so on. So we need to look at it from that perspective.
Thank you very much for your enlightening talk and motivating talk for the students present here, especially those who are going for startups. I want to share that AMU has started incubation center known as AMU IF last year with the funding from the DST, and we are giving seed funding of 10 lakh rupees to every startup company here, and more than 10 companies has already been enrolled and they are working. So the core area of our incubation center is BGEN, that is stand for biotechnology, green energy and nanotechnology. So I will welcome all the participants who want to come to AMUIF and put the proposal. So this was just information I want to share with the students present here. Now coming to your talk, we in AMU have lot of technologies, patents developed by the faculty members and their team, but we are lacking in terms of commercialization of the technology. So any suggestion how we can go ahead for the commercialization of the technology?
Yeah. First of all, it's music to yours. I'm very glad that you started the incubation center, and if you can also bring it under the umbrella of the Bionest incubators, then it becomes a lot more in terms of visibility. Okay, Bionest initially had 3 or 4, now they have about more than 25. You just have to write to DBT and say, this is the incubation center in AMU, and we want accreditation and we want to be a part of the Bionest system. And that helps a lot, because then they take care of the visibility. They have those webinars, they have those industry interfaces and so on. Okay. How to leverage the technologies that are already out here? There are government-owned aggregators. Okay, I can name three. Three is I'm just telling you offhand, but there are many more who do only this job, who pick up technologies from the academia, research institutions at a nascent stage. They scout for partners, industry, and along with the PIs and innovators, and they ensure structuring the entire deal, and they charge a very nominal fee out of the success fee. It's a success fee. You don't have to pay them upfront. Okay. One of them is called BCIL, Biotech Consortium India Limited. Okay, Dr. Purnima is there, and they are very, very receptive. Okay. And the good part is there is competition between these aggregators. Okay, so they will go out of the way to help you. NRDC is one of them. Okay, you can speak to Dr. Amitabh, Dr. Lakshmi Narayan there, and they will. Oh, Dr. Majumdar is there. So NRDC is the second aggregator, and they are good. They are directly under DST. Third is if you're looking at clean energy, agricultural sciences, you said there is a organization called Agri Innovate. Okay, they focus purely on agriculture and livestock and sustainability, green concepts. Three is what I have named, but I can talk to my IP lead, that's Dr. Asia, and she will be able to forward a little more. आप उनके साथ tie up कर लो. वो क्या करेंगे आपके technologies उनके website पे भी display करेंगे. और they come up with periodic expression of interest, and they have a network to whom they send it out. वो customize करके भेजते हैं, and then they follow through to get things done. So I think that could be done.
So on your similar line, your question, a month back, I was in Jensar in Bangalore, Jawaharlal Nehru Scientific Advanced Research. So they presented, we as a team went there, and they pitched with the 5 minutes pitching point. And they all faculties, 4-5 faculties, they came up, they brought their own idea, what is they're doing, what is the data, how they can help. And out of that, we have a couple of collaboration already started with them. Though very small, LCMS based, HRMS based collaboration we have done with them. They have an Orbitrap and all those things. So if that line, your team, if you can make a team and take a lead in it, in your department or somewhere with the life sciences with a different perspective, and make a pitching note and bring each industry people, get 5 minutes, very professional way. I think that's the way people can start.
Exactly. Yeah. Thank you, sir, for that. And one last suggestion, sir. I strongly believe you should connect with C Camp at Bangalore. Dr. Taslim Arif Syed is there. He was specifically pulled back from the US and Europe by the government of Karnataka. 70% of the government grants go to C Camp. अगर AMU और उनका collaboration हो जाता है, and they're open to collaborations, oh, then it'll put you in a different league altogether. Yeah, and Dr. Saab is there at Bangalore, so he can connect you. Yes. Ma'am, बैठ के बात करो चलिए.
Good morning, sir. Sir, I want to know, is there any, today currently, any plant-based vaccines or plant-based adjuvants present?
Oh yes, there's a lot of research happening around plant-based vaccines.
And how to proceed in this, how to proceed?
And the way to do it is, again, what kind of disease are you candidate are you targeting? If it is high potential candidate, okay, then it will definitely get noticed. Then we'll have to look at and what TRL level you are. Okay, this will require a policy change. Okay, so which means we need to work very closely with incubation centers that are operating out of Bioness, because I'm assuming it is at early stages of technology. And then what happens is at every stage the government will handhold you, especially the Department of Biotechnology and BIRAC. They will handhold you and they will take you systematically out there, because there is a very, very strong perception or a trend or rather advice from the ministry to look at plant-based vaccines and adjuvants, although there is no clear policy or guideline around it. So if you have decided to work around it, put up a concept note, talk to DBT, send out a note, they will call you for a discussion, and then go about it systematically. But it is very much required.
Thank you so much, Mr. Ahmed. I think this program will surely lead to great collaborations. Now, thank you everyone. We will now break for lunch. Kindly join us back by 2 p.m. for the afternoon lecture sessions. Thank you everyone. Welcome back, everyone. We will now begin with our another lecture delivered by Dr. Imadul Islam. In this lecture, Sir will walk us through how drug discovery begins, what initiates the process, and who the key contributors are in this complex journey. May I now please call Dr. Imadul Islam on the stage.
Just thinking this will go. Assalamu alaikum everybody and good afternoon. Sorry for being late. We were meeting with your Vice Chancellor, so we cannot leave her. I will request everybody, those who are sitting on the back, please come forward so that you can come closer. That's better. Then you can hear me properly also. I was never backbencher in my life, so I want everybody to be front bencher. Yes, that's the reason that if you are closer to screen, it is better. No, I think that's okay. But they can see, right? Yes, that screen, and I can see this also. So that's okay. That's not a problem. Okay, so the topic of the talk today is art and science of drug discovery. And I will try to set up the stage for next 4 days, 5 days you have. In this lecture, I'm going to talk about almost all discipline. Dr. Azhar Aziz will also talk about some aspect, and Dr. Zainuddin will also talk about some aspect. I'm not going to go into detail. I will just give you a holistic view of the drug discovery in this lecture, and then I have 5 more lectures, and I cover some of the part during that. So let me first introduce myself. My name is Imadul Islam. I work as a Deputy Director of Drug Discovery and Director of Pharmaceutical Analysis in King Abdullah International Medical University Center, Medical Research Center. And before I was in US almost for 25 years or so. I worked in different companies, Bayer, GlaxoSmithKline, Bristol Myers, Lundbeck. And I was, by training, I'm a chemist. I have a PhD from IIT Kanpur and Allahabad University in chemistry. So that's my background, and I have a long experience in drug discovery. A couple of products, they are either going on market or they are in phase clinical trials, 1, 2, 3. So that's how my experience goes along. So the presentation outline will be a little bit different than what usually scientists do. A prologue, monologue, introduction, process of drug discovery, lead optimization, then preclinical phase 1 and 4, and then epilogue, it means last part. This is all we are going to talk in next 4 days, inshallah. So that's the topic. I will just skip that. So I want to give a idea of what we do actually in KMARC. So we do, it is an integrated drug discovery center in Saudi Arabia, and we do target identification. I'm going to talk about that also. Lead identification, lead optimization, then in vivo, ex vivo profiling. Dr. Zain and Dr. Aziz will talk about a little bit, and I will give a little background of development part also. In Saudi Arabia, we don't do any development, so that's why I grade it out. I was just appointed again by the Minister of Health Affairs, reappointed me as a director of PSC, so this is a 2-year term again. I was there for 2 years and 2-year term again. So monologue is, what is drug discovery? I have 40 minutes, right? 40 minutes. Okay, so I just want to see the time so that I don't go over time. inshallah. So monologue is knowledge. What is drug discovery? It is knowledge, experience, and creativity. So this is a page, okay, and there are some dots you can see there. So this is knowledge, okay. Dots are spread on a blank page. But if you have experience, you can connect the dot and it looks like a picture, okay. And if you have creativity, same dots can be connected into a work. You can see a artwork. Drug discovery is not different. It is the same. If I go back, and this is the knowledge, you see all kind of elements are there. Chloro, fluoro, bromo, iodine. Everyone knows that the periodic table, right? If you have little bit background of chemistry, you know that. Then if you have experience, okay, you can connect hydrogen to carbon, it becomes methane. You can connect sodium to carbon, it becomes sodium chloride, which we use every day, eat every day. And if you are creative, then you can connect the proper dot to proper place. तो इस तरह से जो है, you can develop a drug called Januvia, Lyrica, or Leda. So this is the drug discovery in nutshell. The whole parameter will be going around that in next 4 days, okay? So you have to be knowledge, you have to have little knowledge, experience, and creativity. So this is drug discovery. And actually, last part is not showing. So drug discovery is science, art, and it requires knowledge and experience and creativity. So that's the last part which I have written there. Okay, and if you are more creative, then you can see, actually it is not very clear somehow, but if you can little bit visualize that. So the same molecules, they can be like different shapes. With carbon, hydrogen, nitrogen, it can be एक बकरी की तरह से दिखाई दे रही है, एक आदमी की तरह से दिखाई दे रहा है. So this is another, part of drug discovery, that you have to connect these dots. And when you connect the dots, you get a product. So how beautifully Jean-Marie Lehan, he said about drug discovery and the chemistry, that building a legacy with knowledge, experience, and creativity. Atoms are letters, molecules are words, and supramolecular entities like protein. Mr. Syed Ahmad, he talked about vaccines, right? And that is the chapters and sentences. So he was the one who gave this statement, and it's so true these days in drug discovery. Okay, and then the discovery in drug is architect don't discover books. Okay, authors don't discover books. Architect don't discover buildings, and scientists don't discover drugs. So what they do and what we do, we invent, design, engineer molecule to preserve biological system. What is biological system? It is proteins which you are generating in your body. It is creative, collaborative, informed process built on all kind of technical skills and excellence. All of them you are going to listen, inshallah, in next few days. Everyone is here who has done some work. New medicines are not just lying around. Last one is drugs are not magical or propelled. Their behavior should be explained by rational PKPD. So Dr. Zain, who was sitting here, he is a PKPD scientist, and he's going to explain a lot about pharmacokinetic and pharmacodynamic. I wish that we have this screen proper, maybe inshallah. But I'll keep on reading that. So if you do this work and if you discover a drug, better than, okay, yeah, what I see is okay, I'll focus on my screen, that's better. And if you can see there, it is good. So I cannot see here all of that. So top 10 small molecule pharmaceutical by sale, this is 224 data. And you can see that how big numbers are. The Eliquis, there's a one small molecule drug, it is sold 20 billion per year. And somebody has made it, actually it was made by one of my colleagues in Bristol, Massachusetts, and she was rewarded for that. And not only that, this drug is on market, it saves lives. So a lot of people are saved. This is an antithrombotic and blood pressure drug. Similarly, Bactari, Jardine, Tirifata, Farxiga, Artresto, Zentadi, and all those things, you see in terms of billions how big they are in sales. So what drug discovery scientists, they discover these drugs. And they do by chemistry, by pharmacokinetic, and all other parameters, they use that. So this is just, I just showed it there. Numbers are maybe changed, but you can see that these drugs are sold in a very high volume and with a lot of money around. And similarly, you can see other drugs also, so I'll just skip. And then drugs are not only small molecule, there are biologics also. Biologics means protein, vaccines. So like Keytruda, a cancer drug which is sold by Merck is $29 billion. This is like a small country budget, $29 billion, one drug only, one drug. If you discover something in AMU, MU will be on the world map. So just aim for that, inshallah. That's the thing. And this is not very difficult. Dupixent, Drazila, and Opdivo. Actually, Opdivo, I was partly involved in developing that drug in Bristol, Myers Squibb. It was launched when I was there. These are the give back to you, give back to your community that these drugs are available in the market. So with this introduction of drug discovery, I will go to what we are going to talk in next 4 days. The drug discovery process is a target identification, hit identification, lead optimization, and late lead optimization and development. And if you do that, you will earn a lot of money in terms of dollar, and since I'm in Saudi Arabia, I'll say SAR also. So these are the topics we are going to cover, most all three of us, inshallah. And I will talk about target identification, hit, lead, all those things, and what are the aspects of why we do that, and what are the roles for scientists in that, in drug discovery. So drug discovery process timeline is quite long. If I go to next, it is like 6 to 12 years. Yes, I know people are going to ask that AI will help. I will talk about in other topic, that how AI can help. I will not talk today, but this is the process you have to remember, and we are going to talk a lot about this process. So from idea to medicine, it is 6 to 12 years. And the life of the drug is very short, because the patent is only for 17 years and sometimes 20 years. So, you know, you spend billions of dollars, and then you get only very short time. But think back on my slide, where 1 year it was sold for 13 billion. So why not, we should not discover the drug, okay? And this is normal, typical. I worked in a project which was like within 5 years we had a drug already. So this is, I just summed up everything, okay? ये average कर दिया है हमने कि इस तरह से होता है ये. From idea to medicine, 6 to 12 years. So it is a very tedious process. We don't have to get scared, because otherwise we will not have any drug in the market, what we are taking. So company makes a lot of money out of it. And in academic institution like AMU is maybe part of that. So when we do start with that, we look at the patient, why he is sick. Then we try to find out the target, what was the problem with him. And then target identification takes place, gene, protein, Dr. Azhar will talk about a lot. Then I come in picture, active compound, then we optimize the compound, then we go to preclinical, and Dr. Zain will talk hopefully about some PK/PD. Then clinical trial, phase 1 and 2, we will touch about that. I will touch in one of the talk about that how it works and what is the requirement for that, and then registration and market. So drug discovery, so attrition, now, you know, it is not easy, as I told you. So how many compounds usually we need to get a drug? 10,000 molecules. Not always, okay? 10,000 molecules, not always. So hit, we get like 10,000. Then we zone him to 250 molecules. Then next we go to 10 molecules after attrition and after doing studies, one after the other, hit identification, lead optimization. In preclinical, we put 10 molecules. And then in clinical phase 1 to 4, sometimes 4 molecules, sometimes 5 molecules. And if you're lucky, you can put only 1 molecule, which will be really good. And then go to registration, and we get 1 molecule, which is in the market. Okay? So it has a very high attrition rate, and the cost of developing drug is 0.3 to 1.5 billion. Some people say it is more. I'm not going to debate on that. It depends which area and how lucky you are. In my experience, because I worked on many projects, from 500 to 3,000 molecule we made, and we discovered a molecule which was good for some disease. When I give a case study at that time, I'm going to talk about a molecule, a real molecule which is already into phase 2 clinical trial. Okay, so key role of chemist. I told you this morning also I am a chemist, so I will talk more about chemist than other sections. So chemist has a key role in the drug discovery. So you can see in this slide, so there are medicinal chemists who are synthetic chemists, and process chemists are also chemists. I'm not confusing you, but this is like we learn. There is no degree for that. I'm an organic chemist, but I'm a medicinal chemist because I learned at the job, medicinal chemistry. Similarly, a process chemist is an organic chemist, but he learned job at the how to make tons of molecule, tons of kilogram. Okay, think about it. Aspirin is made at least like 179 metric ton every year. So where it is made. So this is the process chemistry job. Then somebody was asking this morning about computational chemistry. Yes, we use computational chemistry also. And analytical chemistry, Dr. Zain will talk some about that. So we use a lot of analytical chemistry to identify our compounds, whether they are right or wrong. And those things are done by all these. So this is the role of chemist in the whole drug discovery process. So they have distinct roles in the drug discovery process, but significant overlap with each other. So I can be a process chemist and I can be an analytical chemist also at the same time. So that works for me. And close communication is required between all disciplines. So here, in lot of departments, industrial chemistry, organic chemistry, and analytical chemistry, they all have to come together to have a company here. So that's very important. So the role of medicinal chemist. Since I'm a medicinal chemist, so I will just give a little bit background. I'm not going to talk about any chemistry, just give you what we do and why you should make a career in medicinal chemistry also. That's another part. So what we do is we design a molecule, novel compound, design synthesis of this molecule, perform synthetic sequence to produce target molecule. So designing novel synthetic route and novel chemistry. We purify the compound, analyze it by spectroscopy data. We register for submitting compound for relevant assays to biochemist. Then designing a better compound, because it is not necessary that you get the first compound which will be really active. So you have to optimize it. Contribute to patent protection and protect the drug molecule. Dr. Rizvi is here. We talked about that thing. It's very important. Don't publish. Dr. Sayyid Ahmed also said that, and I am also saying that don't publish until you patent it if you see there is a potential, because you may be losing $1 billion. And we lost some, in some cases we lost. So talk to people who are knowledgeable in IP arena, and they can tell you whether it is good or bad to publish. Don't publish before you get into that. Any idea, this is not about the molecule only, any device, any sensors, please get in touch with Dr. Rizvi, and he will connect you with the right people. Even you don't have any idea, even you don't have any device, but you have some idea. Ideas are also patented these days. So he will tell you more about that how idea can be patented, and you can patent the idea also without doing anything, but you should have the novel idea. Okay, preliminary route for optimization in conjunction with chemical development. So that's the role of medicinal chemistry. Additional to that, we have some more rule which is called PET reagent or positron emission tomography reagent, which are used for the treatment. Imaging agent, like if somebody has some kind of disease where imaging is required, so we develop, as a chemist, we develop imaging agent also. We develop specific tool compounds for drug discovery also. If you need a compound which can be used for some purpose, we can design and we can at least suggest and diagnostic reagent, which are, diagnostic labs are there. Most of the molecules or compounds or tests which are used there may be developed by the chemist. So that's the purpose of role of medicinal chemist. Okay, so I'll talk one by one, every analytical chemist, what they do. So they support the molecule in drug profile. So when you have drug, if you think you have a drug, so you have to go to an analytical chemist and see whether they are the real compound or something else. So this is the part of their job. And so they develop analytical and spectroscopic method. They demonstrate robustness of method also. And this is all required for filing a regulatory office, like in India it is, I think, a drug authority. In Saudi Arabia it is Saudi Arabia. In US it is FDA. So we have to file, every simple thing they need to know whether what you have done, because they need to know that it is the real compound. Just if you type, just last few days, if you type Professor Kim Janda's name in Google, they proposed a compound 20 years ago, and suddenly they came to know, and he's a professor in UC San Diego. So he discovered that the compound which was marketed for 20 years for at least $120 billion has a wrong structure. Can you believe that a wrong structure was already sold, and he proposed a new structure, and that was right. So it is a big news, actually. Professor Kim Janda, his photo also came all over the media. So it is very important that you should know what you are making. Provide structure elucidation facilities and assure purity and safety of drug. That's very important. Please do not, take it, that it is pure. Just I know it is pure. No, it has to be done properly with the robustness of the procedures and the technique. So this is the role of analytical chemist. Computational chemist, there are several questions were coming this morning, and then afternoon somebody asked me, so I'll just put everything together. Yes, we use computer also. We use computer power to discover drug. We use computer power to optimize drug. So apply computer power to solve the problem, because they can visualize which you cannot see from open eyes. So computer can look into that and tell you that, okay, what you are looking at is right. And usually there are programs which are already known, Schrodinger, MOE. Recently you might have heard about AlphaFolds 1, 2, 3. These are all programs which are done by computational chemistry, and AlphaFold recently won the, 2 years ago, it won the Nobel Prize also. This is a computer program run by Google, another professor from UK. He is the youngest, I believe, 35-year-old, and he won a Nobel Prize by developing this algorithm. So we don't use that, but we use some other programs. We use molecular modeling technique and physical chemistry principle. We calculate energy conformation of drug molecules and calculate binding energy of the drug and proteins. So I work in protein and small molecule both. So these programs are meant to discover drug and to refine your finding, actually. It is not the program which will give you a drug, but it will refine your drug molecule. And you can work in 2D and 3D. There are experts there, and they generate new targets and non-obvious relationship, and they calculate physicochemical properties, which is these days getting a lot of tracking, because physical properties were not earlier used properly. Now they are looking into that also. So this is a computational chemist job in the drug discovery. And we have, you can look into the molecules in 3D and 2D, and how it rotates in the space, how it binds to a target molecule, which is usually protein or ion channels, or GPCR, which is called G-protein coupled receptors. So these are all disease targets, actually. So you can visualize through computers. So this is the task of the computational chemist. So lead optimization. So molecular space is a very big space, okay? And you have to navigate into that, and I will show you what happens once we start doing a drug discovery. So this is a activity X, okay? Then this is a selectivity over Y target. And then acceptable ADME property. So ADME is absorption, disposition, metabolism, excretion. So you have to meet all those, and your molecule should be where? Here. So now how you reach there? So you can see how the space is small, but the task is very big. So you have a molecule which is active on X, it is selective on Y, and then it is acceptable ADME. Everything should be put together and it will go into that small blue place. So we start. So we started with, okay, we got a compound or which I got a protein or a peptide, which is a lead compound, okay? We'll talk about lead in my next lectures. What are the lead compounds? Then we try to optimize it and get to the blue part, but it just goes away and goes to target X. Okay, so now it is out of one part. Now we try again, make more compound, but it does not go to blue part. Then we try to make again, and it goes beyond, okay? Then we keep trying, so it comes into the acceptable, but it does not have the activity X and selectivity at Y. And finally, it is coming closer, and then it goes back again. So this happens all the time in drug discovery. We struggle quite a bit, but sometimes we are lucky, we don't have to struggle, and we get the molecule very quickly. And finally, we get the molecule which we want. So this is the molecule which is important, and that's how the lead optimization takes place. So only a small molecule, a small portion of chemical space has the desired properties or desired balance of properties. The medicinal chemistry's role is to find that molecule that exists in that blue space. Okay, and then and it is supported by all kind of people. I showed you analytical, computational, ADME, molecular biology, pharmacokinetic, pharmacodynamic. Then we get a molecule there. So this is the lead optimization process. And chemical tractability enables us to perform effective lead optimization. So we track the molecule all over. Every time, wherever we go, we track the molecule that it is there. Okay. Now, I hope that you see there. So what I say here that it may not be possible to find a compound meet all the criteria which I told, okay? And it may not be possible for any compound to meet the criteria. It means you have to close the project. So that's very simple, okay? So you do work, and we have closed the many projects, but sometimes we use those projects for other purpose also, teaching purpose also. So that's the drug discovery or lead optimization. So as I told you that I'm going to give you a whole spectrum what we are going to do in the next 5 days. So we reached up to lead optimization. Okay, so in the lead optimization, you need the molecule. You need a molecule in this bottle. This is a vial, okay? We need 10 milligram of molecule. We have to make that. Why we need 10 milligram only? Because Dr. Zainuddin want 1 milligram to see whether it is pure or not, and he will use a method called LC-MS. And sometimes he might ask 5 milligram to dose in the animal. So we need that molecule. So chemist's job is to do a small scale synthesis for identification and characterization purpose. Okay, what happened? Okay, came back. Oops. I think I need some help here. Yeah, presentation mode. Oh, it is here somewhere. Okay, I can go from here. Okay, now it is better actually. It is showing full screen. Earlier it was not showing full screen here at least, but it was showing there. Okay, Now it's better. So these 10 to 15, 10 milligram, and that is where this drug start. 10 milligram of compound you need for identification and characterization. Then using the analytical technique like NMR, IR, mass spectroscopy, HPLC, everything I think I'm sure that Aligarh Muslim University has that in the chemistry department or other departments. And use in vitro assay and enzyme cell-based assay. I think Dr. Azhar will talk more about that a little bit earlier. Why I put my picture there? Can anybody tell? Why not? Because I still work in lab, even today. So that's why I put my picture there, that I was working in lab and somebody took a picture of that, a photo of that. Actually, there was an iconic photo I forgot to bring. Me and Professor Casey, we were talking in a conference room and he got idea. He said, come outside. And we had a coffee cup in our hand and he started writing a structure on that and somebody took a picture and sent it to Professor Bandar Ganavi. And this became an iconic picture in the K-mark that these guys, so I just told Dr. Bandar Ganavi that if you see any chemical graffiti, it means it is me and Dr. Casey. he was laughing. So this is what, you have to be in the lab and you have to work sometime harder. Sometime take it easy. We take it easy. We go for fun. But we wet our hand is still in our lab. So that's my message to you for budding drug discovery scientist. So laboratory scale reactor, this is a process deployment kilo scale reactor. So from 10 milligram, we go to maybe 100 gram to 1 kilogram. Why we go there? Why we directly go to tons? Because then we have to dose in animals. So that is called animal studies. We will talk in our couple of talks, what animals we use, where we use, why we use. We will discuss all those things in the next few talks. This is a reactor. I just want to show you the picture of that this gives you a kilo scale, and we make kilo scale. This may not be GLP material because it is going to animal, but if you are going to human, you have to make a GLP material that is required by FDA. So this is also a chemist's job to do that. Then you can see these are the plant, and Dr. Zainuddin can tell you whether it is from your company or I think it is. I took this picture from your company where you are working right now. Could be. So when we make a tons, multiple kilogram and ton scale, then we go to reactor which are already half buried in the ground, and you won't believe that what you take a small pill is made in about 200 metric tons sometime. 200 metric tons is a lot of drug, and chemists and chemical engineers are involved to make that. And sometimes process is too long and it takes very long to make also. So you appreciate that they are working for the humanity. So now I go to preclinical development. So this is our discovery part. Now you are in drug discovery. There is a function I showed on my first slide that there is a preclinical part also. So preclinical is the CMC, chemistry and manufacturing that requires formulation. You heard today with Mr. Ahmed about formulation of vaccine. That is part of the CMC. And what you use, what you don't use, all those are very, very critical once you make a compound. Whether it will be used by, it may not be amenable to a human being even. So that's very important. And detailed physicochemical property, characterization, impurity profiling, all those things you have to do that. And these are used for animal studies, and data will be used in human phase 1 also. Then benchmark in vivo model. So in vivo model is once you have a compound in vitro, in cells and enzyme, you have already tested, now you have to go to certain animals. So in vivo models are the diseased model sometime, and it is validated model. We use in animal models like mice, rat, rabbit, marmoset, and many times dogs and rabbits also. And then we go to monkeys also. Monkeys are very difficult to do because it is highly regulated, in US at least. In Saudi Arabia, we don't have any facility for monkey right now, but we have facility for up to rabbit and marmoset. So we do that. So ADME profiling, which is done by Dr. Zainuddin, optimize analytical method development. You need a specialized equipment for doing that, basic pharmacokinetic, pharmacodynamic, oral bioavailability, determining metabolism of drug, that's also important. There are many drugs, I will talk some of them, just give you the name that you go and look at and that. And mostly they are done in mice and rats, sometime in non-human primates, which is like monkeys. Then preliminary toxicology, this is also important that you need to take a drug which is safe. So we have to do toxicology and we have to do how much dose you need. You cannot take a drug for 1 gram, maybe you need just only 10 milligrams, so we call it maximum tolerated dose. Repeat dose, non-GLP repeating dose, preliminary cardiovascular safety, pharmacology, all is being done in preclinical side. Several species are used both in vivo and vitro for these works. So this is also a huge amount of work which we usually do to get a drug to market. So human clinical trial, just I will give you a flash of that. So there are phases of clinical trials in humans. So okay, you got a drug, you went up to animal, everything is clean, no toxicity, it is efficacious, it is pure. Now we will start thinking about doing in human. So going from animal to human is another paradigm. तो उसमें कुछ और ही बात होती है, ह्यूमन में जो है वो कुछ और बात होती है. हमने सारा कुछ कर लिया एनिमल में, लेकिन सब कुछ ठीक ठाक दिखता है, बट समटाइम वी फेल देयर. So clinical trials are biomedical health related research studies in human. Clinical trial used to predefine protocol approved by regulatory authorities. In India, it is CDRIR, Central Drug Authority, in Saudi FDA in Saudi Arabia, and FDA in the US. So there are 4 clinical trial phases. One is human pharmacology phase. Second, phase 2 is therapeutic exploratory phase. Third is therapeutic confirmatory phase, and fourth is post-marketing. Let's go by one. So phase 1 trial, first time in human. This is another, if my compound goes to first time in human, I'll be jumping on my feet, okay, that my compound went to first time in human, so we call it phase 1 clinical trial. In this case, we don't use a large number of subjects. They are all healthy subjects. They have to be paid, and actually these days I'm struggling to get human, healthy human volunteers in Saudi Arabia. We need Saudi healthy volunteers, and they are not coming forward because we have to do some trials. So this is a big thing for clinicians that they have to do that. So 10 to 100 individual of both gender. We test safety of the molecule, a small molecule and biologics both. Biologics means protein-based molecules. Determine safest dose, which is dose which has to be used, and we range the dose. The best way is administration, whether it is oral, IP, or any other way. Identify the side effect which are not evident in animal studies. So that's why I said that humans, they showed up some of the side effect they showed up in human. The subject is monitored for several half-life of drug and also determine the volume, distribution, clearance, and oral viability. All will be discussed in next couple of days. In phase 2 trial, done on most result of phase 1, most important phase is drug discovery. Here we use 20 to 300 individual or participant. We continue to monitor safety of the drug, it is safe or not. Begin the access of efficacy of certain disease. All participants receive same experimental dose so that we know it is working. And sometimes phase 2 is divided into phase 1 and phase 2, and that is the reason why we do that. Phase 2A designed to assess the dosing requirement, and phase 2B designed to assess efficacy of the molecule. Phase 3 trial is most important part, and 1 in 10 fails at that stage usually. 1 in 10 drug fail at that stage. So from beginning to here, you already spend about 200 million. Now you can fail or you can make a billion. That's the reason for that. And that's why I put a little bit longer so that everyone can understand. Trial done on larger population from 100 to 3,000. The numbers depend on the therapeutic area also. In cancer, you don't get that many patients. But in cardiovascular these days and diabetes, everybody has at least one of them. Even 40, 30-year, 40-year-old, so you get a lot of patients. That's why the number is there. Can be multi-center trial, assessment of the effect of drug effectiveness, compare the new treatment, which is better than standard of care, side effect. So somebody already has a compound or drug already in market, but we still make because we can be better than them in many ways. Assess risk, outright the benefit, uses two more treatment group, experimental and control. Usually groups are randomized and sometimes blinded. These are the clinical trial part, and the clinician, he knows about that. So we have to do that to understand the activity of the drug on that disease model. Most expensive, time consuming, difficult part of the drug discovery. This takes most of the money. We spend 200 million up to here, and from 200 to 1.5 billion we give to our clinicians. And then new drug application is being submitted for the drug. Now regulatory authority comes in picture, and he is going to look your data from where I started, if you remember, target identification up to this place. Regulatory authority earlier, I remember when I was in Michigan and I saw a truck was going from my company, and I asked, what is that? He said, this is a data for all the molecule, all the studies we did. This is going to Saudi FDA. This is all data, because at that time we used to write by hand everything, and there was no CD and there was no flash drive. So it used to be like that. Now you can put in a flash drive everything and just send it to FDA, and they will approve it. So it's a huge amount of data is being submitted to regulatory authority, which is FDA. Okay, phase 4 trial is not a trial. It is just a marketing and surveillance, whether in larger when it goes, because we tested the compound on 3,000 people. Now you are going to give it to billions or millions of people. Whether it's still there, it does not have any problem. So this is just a monitoring. We call it pharmacovigilance also, in other term. Identify how well treatment works. Monitor long-term benefit and risk. Look for the other uses of drug. And I talked about repurposing this morning. Unsuccessful phase 2 can result in withdrawal of drug from the market. And I will give an example in one of my lecture of why it was withdrawn and why it happened. So this is also an important part of the drug discovery process. So this is my final slide. So what we have to do means to get there. This is a Paul Jensen, who was the chairman of Johnson & Johnson, the parent company of Johnson & Johnson, and he was organic chemist. And I just want to tell you one thing, that when the day he died, he was running a reaction. His reaction was running in the hood, and he was a multi-billion-dollar company owner, actually, but he was still working in the lab. And I have seen a couple of people in Pfizer when I was postdoc there, and they set up the reaction. These are my executive director. They set up the reaction, and sometimes they forget, and they call me, and they say, can you work it up? I forgot. And I said, what it is? And I said, oh, this was this, if you can get something out of it. So people are there. They are not just sitting in their glass offices and they are doing. This person was one of them. That the day he died, he was running a reaction in the end, he died by heart attack. So he says, I always mention this quotation, that a good scientist is someone who succeeds in getting the different scientific discipline to work in harmony with another. And what we talked today with Honorable Vice Chancellor today, this is the same thing, that we have to come together, all discipline in AMU, from engineering college, from chemistry, from INC, come together and just make something happen. Thank you so much for listening. I think I did not over, only exactly 40 minutes. Okay. Okay.
So you can have a, yeah.
I can answer from there if
Okay,
anybody has question. Okay.
yes, sir. Thank you, Dr. Islam, for the informative and well-explained session. Moving ahead, we now invite Dr. Muhammad Azhar Aziz to deliver lecture 2.
I'm here. Okay, thank you, Zainab. I think let's do a little bit of warm-up. Why don't you guys all come in the front, sitting in the back? This will also move your muscles and post-lunch high glycemia. Come on, just rise up and come in the front. Where do I put this? Here. Nobody wants to move, huh? Lunch was heavy? Ah, yeah, it looks like salads only, huh? Okay. All right, so this is going to be the second lecture for today, and I would love to have an engaging class. See, we are doing a coursework, so either we can decide it to be a monologue, like I speak, you listen, or it can be an interactive session. So wherever you want me to stop, you can stop me, ask a question, and let me tell you one thing. No question is a simple or a stupid question. No, every question is a good question. So feel free, because you all are from different backgrounds. आप सबके disciplines अलग हैं. हो सकता है कोई चीज़ जो आपको बहुत simple लग रही हो, वो उसके लिए बहुत advanced हो. तो इसलिए feel free to stop me anywhere, ask a question, and if you don't understand, you can continue, right? Another thing, you'll see some of the slides repeating between my lecture, Dr. Zainuddin's lecture, and Dr. Imadul Islam's lecture. So this is on purpose. Consider it as a revision. Plus, you'll also find something new, new flavor of the same thing. Like he talked about clinical trials, I'll also tell you about the clinical trials. But you will see something additional to it. Plus, it's a revision, so you will remember it better. Right? Can we start now? Nobody moved? So I remember when I was in Case Western, so they used to get up and do some exercise and then again sit down so that you're having full attention. Otherwise, the attention span these days is very low. So keep it engaging. Huh? So I'll be talking mostly from the multidisciplinarity point of view that I started in the morning as well, that it's not just about one man's story. It's a team's story. A team is going to do it. And here's an outline of my content so that you don't get lost. This is how I will navigate. A little bit of introduction, some approaches, methods, phases of drug discovery, drug delivery, some of the introductory slides on that. What are the novel drug delivery systems? Some of the latest breakthrough, AI. Everybody is trying to link to AI. Somehow AI is going to pervade into every aspect of our life. So we'll touch a bit on that as well. And we'll have some conclusion as well. Okay. So we'll start from very basic. What is a drug? Anybody? It's written on the slide, but anybody can answer that. Yes? Okay, good. Anybody else? What is a drug? It could be for prevention as well. So any substance other than food that is used to prevent, diagnose, treat, or relieve symptoms of a disease or abnormal condition. So that's called a drug. And as Dr. Imad said, drug is not discovered, but I'll try to put it in some of the slides that yes, sometimes you're fishing around and you do find the molecules which can qualify as a drug. So drug discovery is the process through which potential new therapeutic entities are identified. So new molecules, new entities, new biologicals, new materials are found from different sources. They can be synthesized in the lab, or you can even get it from a plant. You know about metformin, a very popular drug for diabetes? It's of plant origin. A lot of drugs have got their origin in the plants. Aspirin, cinchona bark. There are so many examples that have got their origin in the plants. And many of them are serendipitous. They were chance discoveries. So they stumbled upon this molecule, they found its potential, and then they qualify as drugs. And the second aspect is the delivery. You found something, it's very nice, very important, very well working, but it cannot go to in the body, it cannot be tolerated by the body, it cannot be delivered to the target organ. So even if you have something, it cannot be delivered, it's a waste. So delivery is another aspect that is as important as drug discovery. So it's a yeah, nutraceuticals are definitely, but they don't qualify as drugs. That's why they don't want approval. They don't need any approvals as well.
So you don't need approval for them.
Yeah, nutraceuticals
But
over the counter.
They are also acting as a drug, food as a drug.
Yeah, but as we know that it doesn't go through the same rigor of drug testing and
that's why it is easier to sell them, sell those things in the market.
Yes, those are nutraceuticals. That could be like a gray area between drugs and
Gray
foods.
area between drug and this.
So drug delivery is a method or process of administering a pharmaceutical compound to achieve a therapeutic effect in humans or animals. Now, why multidisciplinary approach is required? Because it has got different aspects. It involves physicians, it involves biologists, it involves understanding of the human physiology, human body, and a very diverse discipline which is chemistry. They are building molecules in the labs. They know about the synthesis, they know about the pathways of synthesis, but they have got no idea about central nervous system. They've got no idea about the cardiovascular system. So they have to come together. These are, there are so many disciplines that are now getting integrated into a drug development process. So modern drug delivery and discovery is too complex. So it cannot be done by a single person. And you will see in all my slides, this is a repetitive message. Just to break that mindset, which I said in the morning as well, that I am a particular expertise and I can't do that. No, you can. All you have to do is to go and shake hands. That's it. You can do it. So there's a need for genomics, transcriptomics, proteomics, chemistry, AI, pharmacology, and that is how we can increase our rate of success of discovering or finding new drugs. Now, drug discovery timeline, as Dr. Imadul Islam has already mentioned, but do you see a difference? Do you see something new on this slide? Anybody? Anybody tells me the answer gets an extra coffee tomorrow in the morning. कोई तो पता है भाई कोई नई चीज़ है इसमें जो अभी आपने सुनी phase 0 exactly. So phase 1, 2, 3, 4 very popular. Now there's a new terminology that's coming up is phase 0. And you have already seen these slides like what is discovery and then preclinical, which is in the animal models. Then comes the phase 0, which is called preliminary trial. The difference between phase 0 and phase 1 is given here. There are 5 basic differences. It's about the proof of concept is there only in phase 0, whereas there's early data that is available for phase 1. Number of participants are different. And then you also have the method of production of these molecules. It could be GLP, GMP method, GLP is used necessary for phase 0, whereas GMP is mandatory for phase 1. And then you also have got the time span, which is about 10 months in case of phase 0, whereas in case of phase 1, it could be 2 to 3 years. And the results are, of course, different. So it may not be visible to you here, so don't worry, don't bother about writing it all. It will, all these slides from the entire course will be given to you, right? So don't worry about if it's not visible here. So we have got these stages, phase 0, 1A, 1B, where we are testing the safety and maximum tolerable dose. Then we go for efficacy, and then phase 3, phase 4, which Dr. Imad has already told in detail, so I'm not going to repeat that. And the terms that are here, maximum tolerated dose, single ascending dose, this is another model where you keep on increasing the dose and see where is the maximum limit a person can tolerate the dose. And there's a variation of it that is called multiple ascending dose, where you have got multiple subjects. It's not in the same subject. It's different subjects with different ascending doses. So you collect data from different subjects rather than a single person. Now, how do we do this? These are the trials that are coming together, but there are 4 different methods that I can identify and I can classify that how this drug discovery is being done. Random screening is like fishing. You have got a library of molecules, and you are screening for activity, you are screening for targeted effect, and you might be lucky, you might find something. I think the Venn diagrams that you showed, that was excellent. Said that there has to be a molecule which qualifies in all 3 criteria. Then only it qualifies. And it's something like a fishing expedition. You might be lucky. Now it has been facilitated by AI. So earlier there used to be assays. People are running 384 well assays and finding results, and then it was literally a fishing expedition. Now you have got fishing navigation. Has anybody seen the navigation tool for fishes? अब iPhone पे एक ऐप है, उसमें आपको बताता है कि मछलियाँ कहाँ ज़्यादा हैं. So you can go and hook there, and आपका fish catching जो है वो आसान हो जाती है. So same thing is even in the drug discovery. Now with the computational tools, we can narrow down our area of search. So that's with random screening. Then we know a molecule that is already there. We can do modifications. Like we have published a couple of papers where there were iso-indigo dyes. So we did a bromine modification. We added a bromine atom, and we also added a chlorine atom. And we did a modification, and we saw different effects on cell death. So that's called molecular manipulation. Then there's molecular designing, where you're starting from the scratch. You design the molecule, and then and this is also under the rational drug design. So you have got a hypothesis, you know the effects of the chemical entities. So you design the drug up front, and then go and see the efficacy of how it behaves. And then it could be drug metabolized, like there was a drug, but then there's a pathway through which it breaks down. So the intermediates can also work as drugs. So after the drug is metabolized, secondary stuff is formed, और वो भी drug की तरह काम कर सकते हैं. So these could be the 4 methods of drug discovery, right? And so this is a continuous procedure starting from target selection. So you have to identify first, what am I going to target? किस चीज़ को target करना? It could be a molecular target, it could be a cellular target, it could be a genomic target, it could be a transcriptome. Are you familiar with these terms? Genomics क्या होता है? Transcriptomics क्या होता है? Anybody who knows transcriptomics? Genomics? Either the lunch was too low or too high, I don't know. Genomics or transcriptomics? Okay, so genomics is all about DNA, right? And the DNA transcribes into RNA, which is transcriptomics. And then RNA translates into protein, that is proteomics. So you'll see it in my subsequent slides. So once you have the target in mind, then you go for the lead discovery, which Dr. Imad also said about, that you try to find a lead molecule. Lead molecules is among a galaxy of molecules, you identify the molecule which is going to be the most active and most target specific. And it can involve all these synthesis and isolation, combinatorial chemistry, then you have to have an assay to see its activity, whether it's active or not. And we also use a high throughput screening. Medicinal chemistry come into picture where in their lab, they're producing molecules which are having activity, which don't have activity. This is tested later on. And these libraries are available for anybody to buy or get it and use it for their purpose. So there are millions of molecules that are available as libraries which can be tested for their potential as drugs in a particular disease. Then we also have structure-activity relationship studies, in silico screening, chemical synthesis. This all comes under medicinal chemistry. Once you have the compound, we go for in vitro studies. That is what we normally do in our labs. I also have a lab where we do the cell culture studies. It's mostly grown in 2-dimension, or you can also go for 3-dimension organoids, spheroids, etc. Or you can take it to the living models. These are in vivo models, preclinical models, models that are breathing, models that are having life in it. So what comes to your mind when you talk about in vivo models? जब भी ड्रग टेस्टिंग की बात होती है, चूहे सामने आते हैं, उन्हीं पे सबसे पहला जुल्म उन्हीं पे होता है, ठीक है, छोटे चूहे, बड़े चूहे, then we go into even non-human primates. अब खैर वो चीज़ें बहुत बदल गई, so animal models are also very much restricted. Now even FDA is talking about removing all these animal models and having some computational models. कल आप लोगों का tutorial होगा, उसमें आप जाके देखेंगे कि they have replaced the actual animals with the computational based models for testing all this. So I hope you will enjoy that pharmacology models. From there we go into clinical trials. Yes, please. True. Yes, so in computational models, they are even recreating the physiology. I know a group, and I was part of that network in Harvard Medical School, Center for Virtual Tumor. There was no tumor. They developed the tumor as it grows inside the body of a human being. It was having hypoxia, जो center में उसके low oxygen content होता है, उसमें वो भी था, flow of the nutrients. It was also having its growth kinetics, and you can see that physiology on your computer. But that was an effort of a full center, Center for Virtual Tumor. So you can check it out online. So I know you're right that it cannot replicate the physiology of a human being or an animal, but yes, they try to filter it and they try to come as close as possible. So it will not replace, but yes, it will help, it will complement. But who knows? I'm a big proponent of technology. Self-driving cars, it was just science fiction, right? ऐसे कैसे हो सकता है? Self-driving cars कौन बनाएगा? ऐसा हो ही नहीं सकता. But it's a reality now. So coming to the first phase of the slide that we saw, the target selection. This is one of the very important aspects to know what you want to develop the drug against. किस चीज़ को target करना है आपको? In fact, the next course that is coming up, it's all about target identification. कि target तो मिलेगी किसके खिलाफ हमें drug बनानी. अगर वो target मिल जाएगा, तब हमारा अगर सही target हुआ, target से मुराद ये है कि कौन सी ऐसी चीज़ है जिसकी वजह से हम जो disease है उसको reverse कर सकते हैं. जैसे अभी आपने सुना होगा कि कुछ साइंटिस्ट ने स्विच निकाला जिससे कि ट्यूमर सेल्स को वापस नॉर्मल बनाया जा सकता है. So they found a target by which they can revert the tumor into normal cells. So they're not killing the tumor cells, जो कि general approach होती है कि भाई tumor cell इसको kill किया जाए. No, they found a target that can reverse the tumor cell into a normal cell. So identification of target is very, very important. If your target is not correct, I mean, the entire story is going to fizz out. So that's why we can see that most of the targets here, the pie chart that you're seeing here, which has got the highest proportion. Enzymes. So because they are the active proteins, they are the molecules that are affecting the activity of the reactions. So they are delivering there. And the second next is GPCRs, G protein coupled receptors. These are located on the membrane of the cell and they are easily targetable. So we all know about the DNA, RNA protein central dogma, right? तो एक वक्त ऐसा था कि we were proponently believing कि अगर सारी चीज़ों की जो information है वो DNA में है. अगर DNA में कोई खराबी है तो वही चीज़ बनेगी disease. तो everything was in DNA. People zeroed down कि हमें अब solution मिल गया. DNA सही कर दिया, चीज़ सही हो गई. But DNA को target करना is not easy because it's located inside the nucleus. कितने सारे barriers cross करके आने होते हैं. पहले क्या क्रॉस करना है? सेल मेम्ब्रेन, फिर इसके बाद न्यूक्लियर मेम्ब्रेन, पहले पे साइटोसॉल से पास होना है और फिर इसके बाद न्यूक्लियर मेम्ब्रेन एंड देन यू गेट इनटू द डीएनए और डीएनए भी टाइटली पैक्ड, दैट इज आल्सो नॉट एक्सेसिबल एवरीवेयर. सो दैट्स व्हाई द target that is on the surface, which are GPCRs, G protein coupled receptors, they are 7 helix membrane proteins and they are easy targets. And then you also have ion channels, which are also available on the surface, and then other receptors are there. Receptors are all very easily accessible. कि जो दरवाजे पे खड़ा हुआ है उसको गिराना आसान है, जो घर के अंदर है उसको और मुश्किल है. जो घर के अंदर एक लॉकअप में अपने आप को बंद किया वो हाई सिक्योरिटी प्लेस, यू कैन नॉट टच दैट. So in the target selection, we have got cellular and genetic targets, and then we take help from genomics, proteomics, and a lot of data analysis, which comes in the form of bioinformatics. So in case of cellular and genetic targets, this is a lot of basic biology that goes into understanding the disease mechanisms, कि disease काम कैसे कर रही है. जब उसका mechanism समझ में आता है, तो फिर उसके targets मिलते हैं, cellular genetic targets मिलते हैं, right? इसमें receptors, enzymes, जैसे we already talked about that. So we have got the key biological functions पता लगते हैं, and then it could be the disease organism. जैसे अगर हम antibiotics की बात करें, तो we are trying to find out कि बैक्टीरिया के जिंदा रहने के लिए कौन सा जीन important है, कौन सा mechanism important है. So we disrupt that, and then we are able to get rid of that bacteria, right? So we have to understand those basic mechanisms and biology, which can give us the idea about the cellular and genetic targets. But we also are coming up with a lot of efforts, and the data generation has become quite easier in case of genomics and transcriptomics. So now entire genome can be sequenced in about $100. In China, you send out your human genome, it could be $100, $200, $500, you can entire human genome can be generated. So that is creating a lot of enthusiasm in finding out the targets using the genomics or the transcriptomic approaches. And the advent of this technology, CRISPR, so if there is a gene that is solely responsible for causing the disease, we can fix that portion of the DNA and make it healthy. Sickle cell anemia, I think we know about that story. Sickle cell anemia was cured by fixing a single base using the CRISPR-Cas9 technology, which is a gene editing technology. And this is what something that came out of my lab when I was in KMR, that we identified some gene targets by using the genomics approach from the cancer patients. So there were 11 genes that we reported that were novel to be involved in colorectal cancer. So it's coming out of the basic biology. So genomics as well as transcriptomics, both are nucleic acids and they complement each other. But the actual function is being done by the proteins. So the protein guys, they say that no, whatever is in the DNA or RNA, it has to become a protein to be functional, because these are the functional entities. So if you handle the protein, if you tackle the protein, then we have found the right target. So proteomics, therefore, is also gaining traction. Unfortunately, the level of data generation as it is in genomics or transcriptomics, we haven't reached that in case of proteomics, or there's another omics called metabolomics. We are not yet there, right? But all this data is coming out and it's deluging. It's non-informative unless we use these bioinformatics tools. We need to have the computational power to make sense out of this data. 3 billion bases, right? How are these 3 billion bases are arranged? You will know. You send out your genome, you will find out how are they arranged, what is the sequence. But what does it mean? It's like you are given a book in a different language and you are asked, what is it? So you need to have some kind of synthesis method that you can synthesize the information and bring out a target out of that. And that is being done by the bioinformatic tools. And that's why this is a very upcoming field which involves the data analysis, interpretation. It also involves simulation. So like earlier, you have to do the assays. कि drug आपने बनाई और एक target है आपका, दोनों को आपने attach करा के देखा कि activity आ रही है या नहीं आ रही है, उसका कोई output है या नहीं है. But now you can do this computationally. You can do the simulation experiments, and you could do the molecular modeling. You can do the docking stuff. And you can also do the pathway analysis that, okay, if we disrupt this particular step of the pathway, what will be the effect on the end product? And even the clinical trials, especially there's one type of clinical trial I missed here, NF1 clinical trials, where only one patient is there for the trial. And that needs a lot of bioinformatics support. And this is what we are pitching as personalized medicine, that nobody is the same as the other person, everybody is different, and therefore we need to have a personalized medicine approach. This is possible only when we have the power to do a lot of data analysis. Again, coming back to the self-driving cars. Self-driving cars may, you know, what is the main component? Sensors. Sensor उसके ऊपर लगा वो 360 डिग्री डेटा कलेक्ट कर रहा है. सिर्फ डेटा कलेक्ट नहीं कर रहा, उसको रियल टाइम में प्रोसेस करके और फिर इसका इन्फॉर्मेशन भी दे रहा है, and then it's guiding the car for navigation. So it's tackling the situation as well. Cyclist आ गया सामने तो क्या करना है, pedestrian सामने आ गया तो क्या करना है. So this is all possible because now we have the tools and means to do computation. Pehले this was not even possible. I'm not sure if you are from the generation of floppies, but I'm sure you are the generation of GBs, right? You have not heard about KBs and MBs. You have heard only about GBs or TBs. So that's why this bioinformatics-based solutions are possible only when we have gone a lot into our computational power increase. So far, anybody can tell me what is target identification? Yes. You can sit down and tell this. Target identification. What do you mean by target? Okay. Anybody else? What is the target? Does it necessarily have to be a protein? Can't it be it could be DNA as well. Okay, so target is any molecule that, if disrupted, would help in preventing or curing a disease. That could be a simple definition, right? Then we go to the lead discovery. So these are the molecules that are going to be said as the lead molecules, and we have got methods for doing the discovery from plants, or it could be chemical synthesis, as we just talked about, or it could be done computationally. It could be coming from the endogenous ligands, which are having some kind of receptors for disruption in the activity or enhancing the activity. So it's not always about disruption. It can also be about, because like in cancer, you've got oncogenes and tumor suppressor genes, right? So for curing the cancer, you can either enhance the expression of the tumor suppressor or bring down the expression of the oncogene. Both ways. Either you take care of the brake or you take care of the accelerator. Ozempic, we are talking about Ozempic, Mounjaro. Ozempic is an GLP-1 agonist. Agonist means it's enhancing its function. Antagonist is it's inhibiting the function. So it could be on both sides. And this needs to be refined by optimizing these chemical hits for clinical trials, which is known as lead optimization. And this is done through doing some potency, selectivity studies. ADME is absorption, distribution. M is for metabolism. E is for excretion. So this defines, these are the 4 parameters on which we see the efficacy of the drugs, and it also is a qualifying criteria for the drugs to move to the next stage. And oral availability and pharmacokinetic properties, this is an additional thing. Like if a drug is orally available, the compliance is more. Like my boss in US, he was able to develop a oral format for 5-fluorouracil, capecitabine. So when he developed this oral formulation, it was better tolerated and compliance was also better. So, and you know that in vaccines, particularly this oral availability is a plus if it is given to the small infants or kids. I think somebody asked the question about the plant-based vaccines. That's how the plant-based vaccine came into picture. That the promise was that plant-based vaccine will have more compliance and they will be having an oral route. And for the lead discovery, we have got synthesis and isolation step, combinatorial chemistry, and then you have got to have an assay. Assay development is as important. If you can't measure, how would you know? How would you improve? So for that, we definitely need to have an assay. Assay means some kind of a protocol which can tell us about this particular molecule's activity. It could be a color, we'll be talking about this, it could be in the base of color, absorbance, or it could be some kind of a chemical reaction that can allow us to measure the activity of that molecule. And it is achieved through then we go on to the high throughput screening, because we have to screen a number of molecules. So it has to be scalable for high throughput screening, which is known as HTS. Any questions so far? No questions? Okay, so for synthesis and isolation, could be through the separation of mixture. We have got, if you extract something from the plant, is it a single molecule? No, it's a lot of molecules. So you can start with the separation of mixture, then you remove the impurities, or it could be in vitro chemical synthesis. As I said, from the scratch, you can design the molecules, or you can just play around with the atoms for chemical synthesis, or it could be the biosynthetic intermediate, like metabolizing the drug and finding those intermediates. The examples of this is polyketides and alkaloids. Assay development, we just talked about, it's used for measuring the activity of a drug. Without an assay, even if a drug is wonderful, it's going to the dustbin. So you have to have an assay so that you can compare, you can prove its efficacy, and you can prove its activity. And for that, we need to evaluate the expressed protein targets and how is the enzyme substrate interactions are going on. And it also is the kind of filter, whether this molecule should go forward or it should be killed right here. So if an assay cannot be developed for its activity, it could be killed right there. It has to be scalable. For that, it has to have a high throughput screening capability, and so that we can run 384 well plates or 96 well plates. And now this is necessary to have robotic based setup where the human intervention and human errors are minimized, rather it's being taken care of mostly the robotic arms. So then we have got the different assay technologies in high throughput screening. We can have the cell growth based assays that we commonly do in our labs. We grow the cells and see आपने petri plate पे cells को grow किया. You put the compound and see whether the cells are growing more, less, getting killed, or they are alive. The MTD assay. How many of you are familiar with MTD assay? किसी ने MTD assay किया है? Nobody? Okay. Then tissue response. This is functional cell-based assay and also the enzyme test. Enzyme test किए होंगे? Lipase वगैरह chemistry labs में. And the methods that we commonly use for these HTS, it could be spectroscopy based, it could be mass spec like we talked about LCMS and MS and GCMS, gas chromatography or liquid chromatography, or it could be calorimetry based, it could be XRD, X-ray diffraction, or simple imaging based using the microscopy or the radioactive methods as well. How much time do we have? So in the medicinal chemistry aspect, we have got, this is the discipline which is in the intersection of synthetic organic chemistry and pharmacology. And it goes for, and Dr. Imadul Islam has done a lot of medicinal chemistry. He is known as a medicinal chemist. So, and I think your work was on antibody drug conjugates, right? So you have done a lot on those molecules and discovered new molecules or formed new molecules that are in the clinical trials as well. So this is one of the very cornerstones for the entire drug discovery industry that involves drug discovery, lead optimization, process chemistry and development, and this is hypothesis-driven optimization. So if you have got a hypothesis, you have got a target, then it becomes easier than the fishing expedition. So can you see the difference between fishing expedition and target-driven, hypothesis-driven selection? Do you see the difference? एक तरफ आपने मॉलिक्यूल्स बना के रख दिए, अब आप उसको टेस्ट कर रहे हैं कि शायद ये इसमें से कोई एक्टिव हो, शायद इसमें से कोई किसी चीज़ में काम आ जाए, और दूसरा ये कि आपको पता है कि ये मैकेनिज्म है, ये रिसेप्टर है, इसको टारगेट करना है, इसके लिए हमें मॉलिक्यूल डेवलप करना है, इसके लिए एक्टिविटी चाहिए, ये मॉलिक्यूल चाहिए. So that's the difference. So it involves library development, structure-activity relationships, in silico screening, and chemical synthesis. Library development, you can have a lot of chemicals and you play around and get those molecules ready. And whenever you have to start, you can pick up those molecules either through rational designing, you can filter it out initially and then go over for screening. Or if you have the capacity and resources and money, you can have large number of libraries screened. Then you also have chemical synthesis, where you can do the synthesis of your own molecules and do some structure-activity relationship. And this is where the in silico screening comes into play, and that helps. That you do the computer screening of the molecules, and you find out the structures. You see the binding of the target with the drug on the computer, and see the effect on the target as well. And that filters a lot of enormous chemical space. So I think now this is a standard that you do these kind of studies before you go into doing the actual assays, and you do that involves the reagents, the manpower that are going to be a waste. So here I have listed some of the softwares that I use to perform docking and visualization. And so these are some of the softwares that you would find useful if you want to try your hands on. There are more I have just listed here, 6 of them. And we recently had a paper from one of my MTech students where he used this ChemDoc in Vienna and he found the PDL1 inhibitors. So he took the PDL1. Anybody familiar with PDL1? Anybody from the back? Just speak so that I know nobody's sleeping. So PDL1 is a receptor that is helpful in immunotherapy of cancer, which is now one of the most successful strategies. So computational identification of PDL1 inhibitors was done by using these gold and silver nanoparticles, and we were able to conjugate the drugs and see in silico, with the help of in silico studies, that what will be the effect on this particular target. With the in vitro studies, so now once we do the computational studies on the screen, we wear the gloves, we wear the lab coat, come in the lab, and we do the in vitro studies, where we have got the things on the Petri plate or a test tube, right? And this could be the cell lines. How many of you have seen the cell lines? Which cell line? Cell lines are the cells that are derived from the human body or the animal's body, and then they are propagated in the lab. So tomorrow, in the tutorial, you'll also see this snapshot of what are cells, how are they propagated, how are they grown outside the real environment. Or it could be some subcellular components like ribosomes or mitochondria. If your target is in mitochondria, you will definitely need to culture that. Or it could be the extracts, like in case of plants, it could be a wheat germ extract, or it could be reticulocyte extracts. So extracts are giving you the entire cellular contents. So advantages of in vitro experiments are that, first, the biggest advantage I see is you don't have a lot of regulatory approvals needed. You can run as many experiments with the cell lines as much you have. अगर आपको rat से भी करना होगा, rabbit पे भी करना होगा या mouse पे करना होगा, you need to have ethical approval and all that. Whereas in case of in vitro, you don't need any of those approvals. That's the biggest advantage I see. But it's also cost effective. It's faster, like MTD assay is done, you get a result in 24 hours that you have got all the results. But the disadvantages, what could be the possible disadvantages? So it's in vitro. The disadvantage is it's not mimicking the actual scenario. We are trying to mimic the actual human body. We are going to put something inside the human body. So we need to have an environment which is as close as possible. So we gradually go there, and this is far from there. This is only the first step. So how is it done? There are some static models where you have got the 2D cultures, 3D cultures, or spheroids, organoids. These are all called static models. Then you have got the static microphysiological systems. What you're seeing here is now in the microfluidic stages. These are the chips that are available in which you can do the co-cultures. You can have multiple cells. क्योंकि एक तरह के सेल एक जगह पे नहीं ग्रो करते हैं, बल्कि काफ़ी सारे सेल्स एक साथ ग्रो करते हैं. So you can do the co-culturing experiments. And you can also have a dynamic MPS models, which is now set as organ on a chip. So you can have mimicking an organ. Organ has got multiple entities. कई तरह के cells होते हैं, कई तरह की उसमें blood supply होती है. वो सब you can put it on a chip on a micro scale, and that can be used as a model for testing your drug. So this is the one that can be used for assays. जो हमारा lead molecule है, उसके लिए assay development में. Then you also have the in vivo studies. Now we are from the plates and tubes, we are moving to the actual breathing animals. But still they are not humans. बहुत सारी drugs शुरू होती हैं, बहुत अच्छा लगता है स्क्रीन पे computationally. जब हम उसको टेस्ट करते हैं पेट्री प्लेट पे सेल्स के साथ फेल हो जाती हैं, कोई एक्टिविटी नहीं आती है. फिर उसके बाद वहाँ से जो मॉलिक्यूल्स निकलते हैं उनको हम एनिमल्स में टेस्ट करते हैं, वहाँ फेल हो जाते हैं। तो इस काइंड ऑफ फिल्टरिंग शुरू हो जाती है। और बहुत सारे मॉलिक्यूल्स जो बहुत अच्छी एक्टिविटी दिखाएंगे एनिमल्स में, बहुत आपने स्टडी सुनी होंगी, ट्यूमर ग्रो किया माउस में, इंजेक्शन डाला, फौरन श्रिंक कर गया, वाह कैंसर की दवा मिल गई। लेकिन जब वो आता है ह्यूमन ट्रायल्स में, फेल्स। And that happens a lot. That's why the attrition rate for drugs in the pharma industry is very, very high. I hope you'll agree with that. That attrition rate is main cost that we are paying for high, these expensive medicines. Billions of dollars लगते हैं ड्रग को बनाने में, उसमें attrition rate यानी failures बहुत ज़्यादा है, उसकी भी cost जो consumer उसको ही pay करनी पड़ती है। So in vivo studies, we are using the whole organism. It gives us the information about metabolic profile, toxicology, drug interactions. But as I said, there are ethical concerns, cost and time. Each animal is costly, and it also involves a lot of time. It takes months to generate data. Then we have got the different models, animal models for different disease states. You also have the genetic models. For example, in cancer, there's a gene P53 that is mutated, right? So if you are working on something that is having a P53 wild type gene, it's not going to have an actual scenario. So you need to have those mouse models which have got P53 mutation. So this is another branch of another discipline that is working on preparing mouse models, animal models that can ensure that the drug will be successful in the human beings. So I think by now, how many disciplines have you seen that are involved in drug discipline? Can somebody do their memory test? Yes. Good. Bioinformatics, biology. फिजिशियन अभी नहीं आए पिक्चर में, वो आएंगे जब हम क्लिनिकल ट्रायल शुरू करेंगे, वो अभी तक नहीं आए, आगे आने वाले हैं। यस जी, मेडिसिन, ओके, यू मीन द फिजिशियन फ्रॉम द मेडिसिन, मेडिसिनल केमिस्ट, ओके, यस, हु एल्स, माइक्रोबायोलॉजिस्ट, यस, प्लांट्स वाले अंडर रिप्रेजेंटेड हैं यहाँ पे, प्लांट बायोलॉजिस्ट, प्लांट मॉलिक्यूलर बायोलॉजिस्ट, प्लांट फिजियोलॉजिस्ट, जिनको पता है कि अगर पत्ती को आप रब करते हैं तो कुछ एसेंशियल ऑयल्स निकलते हैं उसमें से, और उन एसेंशियल ऑयल से दवा बन सकती है. This is something that only a plant biologist can do. So as we move forward, you will appreciate that there's a lot of disciplines that are coming together for making a drug. They are involved in this. So these are the in vivo studies where we do even the behavioral studies, functional imaging, and we also do the ex vivo studies. I'll come to that. And these are different models, as I said. It could be as simple as a Drosophila. Drosophila is a fly which is very close to have the human genetic setup. And I'll tell you a story about my collaboration. There was a scientist in Mount Sinai, New York. They had developed these Drosophila-based fly avatars. And he made a library of them. So, and you can use them for testing your lead compound. And it was so good that it was something similar to doing a clinical trial. And now it has been taken up by a company in London. They have acquired this technology and they are marketing even in Saudi Arabia because it's highly expensive. And we have got other models like zebrafish and mouse model you all are familiar with. So we have got in silico, in vitro, in vivo, right? So we have gone through all these three things that are used for the assay development. Then there's ex vivo. Ex vivo means outside the human body. So have you heard about CAR-T cell therapy? So the information is taken out from the patient itself, And then those cells are engineered and reprogrammed. For example, their immunity is increased and then put back into the patients to fight. So especially in the case of cancer, and this is highly personalized. Every patient is different. So the patient cells are taken out, engineered, reprogrammed, put back into the patients. Now they are able to fight the cancer and get cured. And this is one of the most transformative therapy in cancer. This is the most promising therapy in cancer. So these are ex vivo models. So why ex vivo studies are used? Because they are the gap between in vitro and in vivo. So they give you the best of the both worlds. You can do it outside the body in controlled conditions, and you don't have to do it inside the body. Like Robert Jarvik. Have you heard about Robert Jarvik? The inventor of the artificial heart. So एक बार वो अपनी कार ठीक कराने के लिए गया मैकेनिक के पास. मैकेनिक पूछा आप क्या करते हैं? कहने में हार्ट स्पेशल. हार्ट क्या होता है? एक हार्ट ऐसी जैसे तुम्हारी कार का कार्बोरेटर होता है ना, वैसा होता है हार्ट. तो कहने तो कौन सा बड़ा काम करते हैं आप? हम तो कार्बोरेटर रोज चेंज करते हैं. तो उन्होंने कहा ठीक है. कार को स्टार्ट किया, ऑन किया, कहने अब चेंज करो इसको. अब इसका कार्बोरेटर चेंज करो. कहेंगे ऐसे कैसे चेंज कर सकते? बंद तो कीजिए इसको, तब तो हम काम शुरू करें. कहेंगे हम बंद नहीं कर सकते. हमें उसी जो है जीते जागते इंसान में थेरेपी करनी है. हमें उसी में हार्ट ट्रांसप्लांट करना होता है. तो दिस इज अबाउट अ लिविंग ह्यूमन बीइंग. उसमें ही हमें थेरेपी करनी है. सो वी आर ट्रीटिंग अ ह्यूमन बीइंग by taking its own material, engineering it outside, and then putting it back. Right? And it's good for personalized medicine. Then we have got, so far we have covered introduction, drug discovery approaches and methods, drug discovery phases. Any questions so far? No? Yes, Shahid. He's one of the students who's very good in asking questions. And you know what is his background? Physics. Yes, Shahid. Yeah. Yeah, his question is कि आप एक ह्यूमन बीइंग में ट्रिलियंस ऑफ सेल्स होते हैं. ठीक है, आपने एक सेल का डीएनए ठीक कर दिया, तो बाकी के साथ क्या कैसे होगा? राइट? दिस इज योर क्वेश्चन. एनीबडी कैन आंसर? जी? Okay. Yes, so usually जो इंजीनियरिंग की जा रही है वो उन सेल्स की जा रही है जो कि पूरी बॉडी में सर्कुलेट होंगे. राइट? तो वो जहाँ पे फैक्ट्री है, जो बोन मैरो है, वहाँ पे चेंजेस किए जाते हैं ताकि जो फैक्ट्री से जो नए सेल्स निकलें वो सब तरफ हेल्दी. फैक्ट्री में जैसे चीज़ ख़राब बनेगी वो सब जगह ख़राब पहुँचेगी. फैक्ट्री में सही कर दिया, सब जगह सही पहुँचेगी. Okay, so now we'll just look at the data-driven, because I'm reinforcing the message of multidisciplinarity. Multidisciplinary approach, we have got the candidate molecule in silico, then we have got this pharmacokinetics, safety, efficacy, and all this is now getting a boost by using the AI, by using simulation, by using experiments. Now once the drug is made, the next big challenge is its delivery. अब इसको पहुंचाना है अपने टारगेट तक. आपने बहुत अच्छा सा खाना बनाया, but it's not reaching to the right person in the right format. Zomato पे ऑर्डर करते हैं ना, बहुत अच्छे मेन्यू में चीज़ें मिलती हैं, दिखाई पड़ती हैं, but if it's not reaching the person in the right format, it's spoiled. So the purpose of doing drug delivery studies is to let the drug reach to its desired place in the right format, in the right dose, in the right time, and also getting cleared off उसको बॉडी में रहना भी नहीं लंबे वक्त तक. So it should be excreted out as well. So that's the whole purpose of drug delivery stuff. So drug delivery is through all the routes that can absorb. It could be oral, buccal, pulmonary, through the lungs, and there are different formulations for different routes of delivery. जैसे कुछ ड्रग्स हैं जो कि इनहेलर के फॉर्म में हैं. कौन से ड्रग्स जिनको कि लंग्स में जल्दी से आना है, राइट? कुछ ड्रग्स हैं जिनको ब्रेन में पहुंचाना है, कुछ ड्रग्स हैं जिनको स्किन में ही रखना है. तो यू डोंट नीड टू टेक इंट्रावेनस. You will need something that is applied on the skin. Then there are nasal, sublingual, depending on the target organ, it could be different. And the one that is going to be everywhere will be the intravenous through the blood, because blood is going to reach everywhere. So these, there are different formulations, there are different materials that are being used for the drug delivery system. Liposome, it's a ball of lipids. Ball of lipids helps to protect as well as go and fuse with the cell membrane, because cell membrane is made up of lipids. So it can go to the cell, fuse and deliver easily, right? They are same. So liposome, dendrimers are the other stuff, inorganic nanoparticles, lipid nanoparticles, hydrogels, carbon-based nanoparticles, magnetic nanoparticles. These are all packaging. ज़्यादातर इसमें से जो है सब पैकेजिंग है. कैसे पैक करेंगे? जैसे कुछ चीज़ों को पैक किया जाता है कार्डबोर्ड में. कभी आपने ऑर्डर किया हो तो कुछ नाजुक चीज़ें जो हैं वो बहुत अच्छी पैकिंग में आती हैं, बहुत उसमें कई मल्टी लेयर्ड पैकिंग की जाती है, राइट? So in this case also there are different materials for different types of drugs. There could be water-soluble drug, there could be the drugs that are water insoluble. For that we need to have a different strategy. They are not soluble in water. So liposome-based drug delivery, this is what it looks like. It's a ball of lipids and this has got a core of water. But there are variations of this which are known as solid lipid nanoparticles, which have got the ball, which is where the core is also made up of lipid. So you can pack the lipid soluble drugs as well. Although it is desirable that we have water soluble drug, but somehow it's not becoming water soluble. So we can change the formulation. Packaging बदल देंगे. अगर packaging हमने उसकी कर ली, then we can use it as a drug by using these drug delivery systems. And then we can add some of the addresses, the peptide, for making it specific for a particular receptor, or we can use polyethylene glycol for protecting it from the immune system, or we can add the DNA or antibody for making it more specific. Next is dendrimers. Dendrimers are branched structures. This is a branched structure, and these branches are all having chemical groups that can serve as the attachment point for the drugs. So dendry means tree. So it has got a tree-like structure, और tree की हर branch पे आप drugs को load कर सकते हैं, और load करके then, so they are based on how many circles are there are generations. So if you have got only one circle, G0, two circles, it's G1, G2, G3. So far G4 तक लोगों ने dendrimers बनाए हुए हैं, right? And it has been used for drug delivery in case of eye formulations. So I'll try to zip through quickly. Then we have got the hydrogel-based drug delivery system. Hydrogel is a material which can absorb a lot of water, so it can swell up. So it is in its powder form, it's dry, but as soon as it gets in touch with water, it can swell up, and it can also release on reaching a particular target based on the difference in pH or temperature. So these are the hydrogel-based drug delivery system. I think you'll see, because this is an introductory lecture, you'll see more in detail in the upcoming lectures. So examples are given here, and we can go through that in detail. Inorganic nanoparticles are there, which are used for crossing the blood-brain barrier. Blood-brain barrier, it is a natural mechanism to prevent the unauthorized passage of molecules into the brain. It's a very tight junction where not everything can pass into the brain. So for passing through that barrier, there are certain inorganic nanoparticles that can be utilized, and they can go through that. And it's also for the ocular drug delivery for the eyes, cancer theranostics. Theranostics are the agent which can be used both for therapy as well as diagnostics. Those are known as theranostic agents. Then we have got the carbon base, so we have got inorganic systems as well as carbon base. So people have used carbon nanotubes and even the fullerene balls for packing the drugs and delivery as well. So they have all been utilized. They have been utilized in photothermal therapy as well. So I'm not sure how familiar you are with these terms, but I'll be happy to answer, and it may come up in the subsequent lectures as well. Then we have got the mRNA delivery system. I particularly emphasized on this because this is an upcoming field in case of the drugs. mRNA, the COVID-19 vaccine, everybody knows about. It was an mRNA formulation. Some of the companies, they have got the mRNA formulations, and they were protected in these liposomes and delivered. Then we also have the magnetic nanoparticles, which are easy to locate, easy to trace, and also they are being used for the drug release by the application of the magnetic field. And if you come to the recent advancements, I just wanted to mention this guy who's got the Nobel Prize, David Baker. So I'm switching gears into the AI again. David Baker, they recently designed an antibody on the computer. अभी तक एंटीबॉडीज बनाने के लिए एंटीजन को आपको एनिमल में डालना है, फिर इसके बाद इसमें से जो एंटीबॉडीज निकलेंगी उनको फिर स्कैन करना है, देखना है किसकी एक्टिविटी है, कौन सी न्यूट्रलाइजिंग है, कौन सी नहीं है. बट डेविड बेकर की लेटेस्ट रिपोर्ट ये है, थिंक 2 weeks ago it was published that they designed an antibody from scratch on the screen, and it was found to be active. Another upcoming field is click chemistry, which also got the Nobel Prize a couple of years ago, which is a green method of making molecules, and it's very helpful in designing new molecules that could be served for the drug purpose. So we have got drug discovery, drug delivery, and it has to be combined with some bioimaging and material science for taking it to the public. And this is being aided by AI again for data processing as a lab application, and the self-learning models where the data generated and data applied teaches its own model. I think that there's key applications of AI in drug discovery. In all the stages that you have seen, AI is now helping. I would put that in a simple way, right? And there are a lot of benefits. I think we need to have a fresh morning session to discuss that, because many of you would disagree that AI is not always beneficial. This has got this. So I'll leave it for this would be my second last slide, and this will be the conclusion. So all these slides will be provided to you, so don't worry about that. And if you have questions, we can discuss it tomorrow. So no questions? Yes. Either you have to come forward or speak a little louder. Yeah, see, vaccine is a type of drug. So it could be using the same mechanisms that we are talking about as drug delivery, like liposome based. It could be the same. They could be oral as well. They could be oral, they could be intravenous, it could be intramuscular, they could be intradermal, subcutaneous. All formulations are there. Ji? Polio is oral, yeah. That's a very popular example. All right, so we'll conclude here. I hope you had a learning and enjoyable session today. Tomorrow we are meeting at 9:30 in the civil engineering department. How many of you are familiar with that place? If not, we can show it to you now. Rest of you are not familiar? सबको पता है civil engineering कहाँ है? अगर नहीं पता तो please अपने colleague, otherwise we'll post the location also on the group. ठीक है? तो we'll meet there at 9:30. फिर दो groups बन जाएंगे. एक group जाएगा pharmacology, वहाँ पे आप लोग को tutorial दिया जाएगा, और एक group जाएगा nanotechnology center, वहाँ पे tutorial होगा. Then we'll switch. बस arrange कर दी है, but I love to walk. अगर आप लोग walk करना चाहें तो it's a very beautiful campus. You can get to see. It's optional. वैसे बस available रहेगी. It will be there. और कोई announcement नहीं? All right, and so I'll see you tomorrow then. आज की सबकी attendance लग गई है? Because attendance is mandatory, we have to send the attendance to the GIAN Ministry of Education. कल भी ख्याल रखिएगा अपनी attendance का. Used to my next stuff.
I don't know. Ask in the audience. I don't know.
क्यों?
It's attendance sheet. It's a registration sheet. I request the participants to mark their today's attendance tomorrow, okay?
Honestly.
Yeah, please honestly. Okay. Thank you everyone. Also, coffee is also available outside, so you all are requested to go there.